Dr. Barney Graham. Thank you. So next speaker on the list is Dr. Barney Graham. He's Deputy Director of and Chief of Viral Pathogenesis Lab at National Institute of Allergy and Immunology Division, Vaccine Research Center. I mean, we're very fortunate to have a polymath, somebody who's a physician and a basic scientist. He had his bachelor's from Rice and MD from University of Kansas and PhD from Vanderbilt University. He's been involved in a host of viruses. He's really an amazing, virologist. He's worked on HIV, Ebola, chikungunya, RSV, influenza, Zika, you name it. You know, he's a Dr. Virus specialist, and so therefore we're really privileged to have him speak to us. And Barney, thank you so much for joining us this morning, and we look forward to your talk.
Good morning, and thank you for the opportunity to be here. And I'm going to be talking today about rapid development of coronavirus vaccines and try to put it in some historical context. First, I come from the Vaccine Research Center, which is on the campus in Bethesda at NIH. This was a center established about 20 years ago for developing an HIV vaccine. And even though we haven't been successful in that mission, we have used the technology derived from that effort on a number of other viral diseases and unmet medical needs, including emerging infectious diseases listed here. We can do this because we're privileged to have not only this basic research building, but a number of engineers in process development and in pilot plant manufacturing, a self-contained clinic for vaccine evaluation, and a laboratory dedicated to GLP evaluation of our clinical samples. It's directed by this group of PIs and program heads, and our leader, John Muscola, is shown here in the middle. Over these last 20 years, we've delivered vaccines by a number of platforms, including nucleic acids, which I'll talk about today, vector-based approaches, as you just heard from Dr. Schutemacher, and virus-like particles, for instance, for chikungunya, proteins that I'll mention for RSV, and nanoparticle displays that I'm sure you'll hear about later from Dr. King for influenza, and monoclonal antibodies. For instance, we've sent some of these all over the world. MAB 114 that was used to treat Ebola effectively in 2018 in Eastern Congo is an example of something that came from the VRC. So coronaviruses are a large family that have a large animal reservoir, and it's likely that we'll continue to be under threat by these over time. We have 4 endemic coronaviruses, but in 2002, we experienced the first SARS, which infected a little over 8,000 people and had a 10% mortality. Fortunately, it died out. And MERS came about 10 years later in 2012 and 13 in the Middle East, and in this case had a 40% mortality. Fortunately, the current virus is doesn't have that high mortality rate, or we'd be in even more trouble. It's likely that these will continue because, as Ralph Barrick predicted even back in 2016, there's a large reservoir of SARS-like viruses in bats that are poised for human emergence. For instance, this WIV1 strain that he discussed 4 years ago. This pandemic is not the largest infectious disease disaster that the world has seen, but in modern times, it's really second only to HIV, which has now killed upwards of 35 million people. This pandemic is now larger in terms of mortality than the recent historical influenza pandemics going back to the 1950s. It is not as bad as the 1918 flu pandemic, but this was a pandemic that occurred prior to modern medicine and even prior to knowing that this was a viral disease at the time. So at the moment, we're still climbing in mortality globally. As you see, these are infection cases over 500,000 per day and climbing. It's probably going to get worse up to January. And the deaths are steady at around 10,000 now and climbing. So this is something that we have to move quickly. And so I want to start by asking the question, why should we vaccinate? The goal is to have an immune population, and that's either going to happen through infection or through active vaccination. And the problem with letting this occur and generating population immunity through infection is that we're going to lose a lot of people, as we already are. With vaccination, it's a much safer approach to generating an immune population. And the reason we need population immunity is, I try to illustrate here. In a non-immune population, you're going to have a very high transmission rate. In an immune population, the susceptible people, the blue people here, are in essence socially distanced, and you'll have much lower transmission rates, hopefully much lower inoculus size. And the consequence of that is high severity of disease in the non-immune population and milder disease among the immune population. So we need to vaccinate, and we need to do it quickly. And fortunately, during this pandemic, there's been a huge global response. WHO lists these new vaccine development programs on a landscape slide, and there are already 47 vaccine candidates in clinical evaluation around the world using a number of different technologies, not just the gene-based vectors, but protein-based approaches, peptides, virus-like particles, chimeric viruses, or even whole inactivated viruses. And there's another 155 vaccine candidates in preclinical evaluation. The 4 starred approaches I have here have already advanced into phase 3 clinical trials, which is unprecedented even in modern times, to have vaccines into phase 3 testing within a year. And so why are we going fast? Why do we need to go fast? I tried to review some of that in this commentary early on, and it has to do with how epidemic curves are represented here with either infections, hospitalizations, or deaths respond to interventions. And so if you have this as the epidemic curve, if you can respond early, even with a relatively poor vaccine, in this case, 40% example, you can drastically attenuate the curve of the epidemic. And that actually is more effective overall than responding with a better vaccine at a later time point. So getting vaccines into the population, into the epidemic as soon as possible will, even if they're not as good as they may be years from now, is a much more effective approach to deal with this pandemic. So we have tried to go fast with our vaccine and monoclonal antibody development program. This was made possible largely because of prior work we had done that I'll explain a bit later. Starting on January 10th, when the sequences were posted in a collaboration with Moderna, we started ordering sequences and initiated GMP manufacturing without any additional experimentation based on this prior work, and were able to get this mRNA 1273 product into phase 3 in about 6.5 months and into phase 1 in about 65 days. Likewise, with the monoclonal antibody development, which in many ways is a byproduct of the vaccine program, because the same reagents are needed to isolate B cells from convalescent donors and then develop and screen for high neutralizing potency. In collaboration with AbCellera and Lilly, this MAB555 has advanced into phase 3 within 5 months and is now has been granted an emergency use authorization for its what's now called bamlanivimab. So we think that our experience over this last 10 years has suggested to us that from all the pandemics that have occurred, from chikungunya in 2012 and MERS and Ebola in 2014, Zika in 2016, et cetera, that not only do you have to go fast, but you have to do something that has some precision. And so we are using platform technology in collaboration with Moderna, the mRNA platform, to go fast, but we based most of our antigen design approaches using atomic level structure. So structure-based vaccine design is the approach that we take to make more precision antigens. And in this case, we've used a prototype pathogen preparedness focused mostly on the MERS coronavirus over these last few years. A lot of this goes back several years to this finding we had with respiratory syncytial virus, which has been with us since the mid-50s and has had multiple failed vaccine attempts. And during the period between 2010 and 13, we were able to capture the structure of this pre-fusion conformation of the F-glycoprotein. This was done in collaboration with Jason McClellan, who was a postdoc in Peter Kwong's lab at the time. And the important finding from this basic research project was that there was a new neutralization sensitive epitope at the apex of this pre-fusion conformation. And when this molecule, as it does, spontaneously flip, even on the virus without triggering by a host cell, you lose this epitope. And this protein and this conformation over the last several decades has been used in vaccine trials with similar results of boosting neutralizing activity around 2 to 3-fold, which has not been enough to advance it as a vaccine product. But if you can stabilize this molecule in the pre-fusion conformation by adding a C-terminal fold-on trimerization domain or internal disulfides and cavity-filling mutations, you can now achieve a 16-fold or greater boost in neutralizing activity from a single dose, even without adjuvant. So now this is a vaccine candidate that has some promise of advancing. And just to emphasize the importance of this concept is I'm showing you a cartoon that illustrates how class 1 fusion proteins operate. They are required for viral entry. They begin on the virus membrane in one conformation, undergo a dramatic rearrangement that grabs the host cell membrane, creates this fusion event to allow the host genome to enter the host cell. So without this protein function, the virus cannot infect cells. So attacking this is a major strategy for making vaccines against envelope viruses that use class 1 fusion proteins. And this includes coronavirus. So even though some of the domain structures are a bit different, and if coronavirus has an S1 component cap that pops off before this fusion machinery operates, all of these class 1 fusion proteins are designed in a similar way with an endoproteolytic cleavage site that exposes a hydrophobic fusion peptide, and then these heptad repeat regions are what holds the membranes together to create the fusion event. So The vaccine strategies against these proteins can also be similar. So solving this atomic structure of F revealed a new target of vulnerability. We now have proof of concept for structure-based vaccine design that preserves neutralization sensitive epitopes at the apex of these class 1 fusion molecules, and we think that they can be generalized across other virus families, as we've done with paramyxoviruses, but also now with coronaviruses. So the MERS outbreak was happening around the time of this finding the prefusion structure. And so because it seemed apparent that another beta coronavirus was likely to emerge, we concentrated on solving the spike structure, and we're not successful with MERS or SARS, but in collaboration with Jason McClellan again, and now with Andrew Ward at Scripps, we're able to solve the structure of this endemic beta coronavirus, HKU1, because it had inherent stability and allowed cryo-EM structure analysis. And we learned a lot from this. Not only is it a very unique protein with this interesting mobility of its receptor binding domain that is required in order for it to bind its host cell receptor ACE2. But we found that a stabilizing mutation that goes, as Hanneke mentioned, between the central helix and the heptad repeat, which is a hinge region for this fusion machine to work. If you put two prolines there in the same position, not just for HKU1, but for other beta coronaviruses, you can largely stabilize the molecule. So not only do you then preserve the neutralization sensitive epitopes at the apex of this molecule, but it also, especially important for gene-based vectors, increases expression levels. So for instance, in MERS, it's 50-fold higher in the 2P stabilized version than it is in the wild type version down here on size exclusion chromatography. So knowing that and wanting to advance this prototype pathogen concept with the platform manufacturing approach, we started collaborating with Moderna in 2017, working both with Nipah for paramyxovirus and MERS coronavirus for the coronavirus family. We were able to show that very low doses of mRNA delivering a stabilized spike protein, even down to 0.1 microgram dose levels, could protect mice from infection of the lung in this human DP4 transgenic mouse model developed by Ralph Barrett. In this case, both the 0.1 and 1 microgram dose levels completely protected against weight loss, but even in the group that had breakthrough infection at 0.01 microgram dose level, there was the partial immunity resulted in partial protection. There was no evidence of enhancement, and over the course of these studies, we've seen no evidence of enhanced disease, as Hanneke mentioned during the breakthrough infection from an mRNA approach to vaccination. So we know that proline stabilizes prefusion S and that these stabilized trimers are more immunogenic. We thought it could be a general solution for beta coronavirus vaccines, and we had shown that mRNA was a suitable and rapid manufacturing approach for this type of virus. And then 2020 hit, and everyone knows now that we had SARS, and as I said earlier, it's not that surprising. We have these 4 endemic coronaviruses, 2 alphas and 2 beta coronaviruses, and these viruses have been entering the human population now for hundreds of years. And given that we've had 2 beta coronaviruses in the last 20 years, it wasn't surprising that we had a 3rd one during this last year. And this is a beautiful scanning electron microscopy from UNC, and of the SARS virus infecting a ciliated respiratory epithelial cell. And you see this panel here is enlarged on this side, and what you see is large numbers of spherical viruses clumped together, probably held together with mucus. And just recolorizing, you can see that these big bumps on the surface of this virus is the spike protein. And so it's easy to see why this should be a target for antibody and vaccine development. Because the way this works, as I showed earlier, is that the spike protein, which sits on the virus surface, is looking for a human cell with an ACE2 receptor. And it can engage that receptor when the receptor binding domain is in the up position. And if you can elicit antibodies either to the RBD or the N-terminal domain or even the stem of this molecule, you can interfere with its fusion machinery and prevent viral entry. So that is the goal. And when these sequences came out, our philosophy is that the protein is the beginning of everything. And so in collaboration with Jace McClellan, again, we were able to make a protein to solve the structure of this spike molecule. And once you can see that it's in the right conformation, it allows you to have the confidence to rapidly make diagnostics, as we did with CDC, to probe convalescent B cells, as we did, as I mentioned, with Lilly and Epsilera, and to make vaccines as we have with Moderna. And just to illustrate the importance of this, of using a pre-fusion stabilized spike, these 5 out of these 6 groups that are either making advectors or protein-based approaches or nucleic acids are using a stabilized version of the pre-fusion spike protein. So for Lilly, I mentioned that we had isolated this antibody 555 that's now called bamlanivimab, and a couple of weeks ago, this was published, their study in which they used the antibody to treat people with mild or moderate symptomatic coronavirus disease. They were able to show not only a rapid decrease in virus titer, but prevention of progression to hospitalization or ER visits. Placebo recipients were progressing at about a little over 6%, and the treated groups were at the 1.6% range, so about 72% reduction in disease progression. And that is now led to the announcement that they've been granted emergency use authorization for this antibody to be used to prevent progression of disease in people with symptomatic coronavirus. For the vaccine, over these last few months, we've been able to show that it's immunogenic in mice, monkeys, and humans. Monkeys, we can generate with a 100 microgram dose after 2 doses, about 3,500 nuc titers with the live virus neutralization assay. And in humans, at 100 or 250 microgram dose, we're well above about 3 to 5 times higher than the GMT of a pool of convalescent sera shown on the right. We've been able to show protection of both the lower and the upper airway in mice and in monkeys. And we've been able to show that it has a safety profile that we think is favorable with a Th1 biased response in young adults, 18 to 55, but also in older adults where the same Th1 biased response is seen even in people over 70 years of age. And fortunately, as suggested in the earlier talk, that these responses are similar in the older age groups as they are in the younger adult age groups. So these people who are about, on average, 40 years older than this group has a very similar neutralizing titers in the range of 1,000 with this live virus neutralization assay. And we were encouraged last week when the Pfizer vaccine was shown to be 90% effective, in part because this vaccine is designed very close to the way the Moderna mRNA vaccine is designed. It has the membrane anchored S spike protein with the 2 proline stabilizing mutation. It uses a modified nucleotide 1-methyluridine, and it has a very similar lipid nanoparticle formulation that encapsulates the product and facilitates both stability and entry into cells. So we will find out in the coming days about the efficacy for the Moderna product. I mentioned the Lilly product now has emergency use authorization. I mostly want to stress the importance of fundamental, basic, and translational research that created the knowledge base and the information needed to do this rapid development that involved both precision and speed, and also the importance of pre-established public-private partnerships that made this work possible. And I'll stop here and thank my colleagues at the VRC, the PIs and program heads, Jason McClellan, who's been involved in all of our work from RSV on through the coronaviruses, Dr. Kazmikia Corbett, who's sort of hiding here, but has led our small coronavirus team over these last 4 or 5 years, and Moderna for the RNA, Mark Dennison at Vanderbilt, and Jim Chappell for the neutralization assays, and Ralph Barrick for the murine studies. DMID, another division at NIAID, helped do the phase 1 clinical trial. And I mentioned Abceller and Lilly, and these are other collaborators who've helped make all this possible. And I'll stop. Thank you very much.
Dr. Graham, thank you very much. I mean, really inspiring talk in terms of where the future is going. And I like your, strategy tremendously, which is seen to have perpetuated through all the virus anti-vaccine efforts. This is great. The proline strategy is fantastic. Yeah, thank you. So I want the audience has questions. Let me pose the questions first to you. Mark Oldham asks, is the choice of a delivery mechanism influenced by COVID being disease of the endothelium, and would the chosen delivery mechanism potentially need to be different for different cell types? If they are the primary targets.
Right. I think the question is about whether these parenterally delivered vaccines are going to create sufficient mucosal immunity in the future. And the initial vaccines, in part because of the need for speed, are being delivered parenterally. And the goal here is to generate high levels of IgG that's neutralizing, that should transudate and protect the lower airway and the lung. It will also penetrate the upper airway and probably reduce virus peak titer and reduce the length of shedding, but it may not entirely prevent infection of the upper airway. Eventually that will be good because it'll help boost immunity periodically, but in the near term, it's going to require us to still maintain some caution.
Yeah, thank you. Yeah, the second question of the same gentleman, which is, Eli Lilly antibody treatment is approved only for adults over 65. So it's not recommended for patients who have been hospitalized or on oxygen.
Right.
He's asking the question, do such restrictions result from, what's the origin for these restrictions? That's
right. Well, first, in my experience, it's very rare for an antibody to be effective given after infection of a respiratory virus. So in part, it's surprising to me that the antibody has worked very well at all. And I think it speaks to the relatively slow progression from upper to lower airway infection and relatively slow disease progression in this particular virus that it works at all. So this antibody treatment does not work in people who have severe disease hospitalization. It does work early in infection, similar to the way an antiviral for influenza works when you use it as early as possible. And it is approved for not only people over 65, but anyone over 12 who has another risk condition. So if you are over 12 years old with a risk factor that suggests you're at risk for severe disease, it could also be used. Ideally as an outpatient or at the very beginning of a hospitalization.
Great, thank you. There are many other questions on the Q&A, and if you have time, feel free to answer them, especially this follow-up questions on the endothelium question that was posed. And so by the time we can get Maria to load, maybe you can answer that question. He says that, what about peripheral blood and tissues which are not easily accessible for assay? Do you see what's the response to the vaccine in those tissues?
Right. Yeah, mostly we can assay peripheral blood, except in our non-human primate studies. Of course, we can get to the lung tissue itself. And ultimately, what you'd like to have is not only high levels of neutralizing antibody, but I think tissue-resident CD8 T cells that reside in the lung tissue. You may not be getting these from parenteral vaccination, and so there's still going to be some work to do after this first generation of vaccines to get it as good as we can.
Fantastic. Thank you again, Dr. Graham. I mean, Bonnie, it's a really spectacular talk. We appreciate your, talking to us this morning. It's great. Now it's my great pleasure to introduce our next speaker, Dr. Maria Elena Bottazzi, who is Associate Dean for the National School of Tropical Medicine and Professor of Pediatrics and co-director of the Texas Children's Center for Vaccine Development at the Baylor College of Medicine in Houston. Dr. Bottazzi got her degrees from Honduras, and she got her doctorate from University of Florida. She's been one of the pioneers in terms of the vaccine development. She's a member of the National Academy of Science of Honduras. She serves as a co-chair for the New Vaccines and Therapeutics Division Task Force for the Lancet Commission on COVID-19. She's a fellow of the American Society of Tropical Medicine and Hygiene and on the executive leadership team in academic medicine. And so, Dr. Bottazzi, it's really a great privilege to have you talk to us, and we look forward to your presentation. Thank you.
Thank you. Thank you so much, Dr. Shankar. And hello to everybody in the audience and my colleagues that already had presented. I think it's a good segue. And so today I would like to speak to you a little bit about our story. I'm calling it a little vignette for you on how we at Baylor College of Medicine and at Texas Children's have been able to advance certainly coronavirus vaccines, but now specifically COVID-19, and our recent agreement with Biological E, which we have some very interesting advances. So I just wanted to, again, pass through my disclosures and remind everybody that, as you know, if we keep track of all the different trackers, there's certainly, this has been an unprecedented number of groups and initiatives, and I think all we have the aspiration to very soon have yet another tool to add to the toolbox to try to really start impacting the vaccine as another method of preventing this disease and eventually even interrupt transmission. I just put this slide mostly because it shows the ones that are the most advanced in development, but as you can see, there are many others that are coming behind, not only in phase 2, but also in phase 1. And I think the aspiration is that on aggregate, we're gonna be able to determine which of these vaccines.
I'm sorry, you're not in a presentation mode. We are not seeing a slide move. Can you go to the presentation mode?
It is on presentation mode.
We are seeing the slide view with all the slides, and it's not advancing.
Let me stop sharing and start again, maybe?
Yeah, please.
Because it was advancing on mine. I hope it's not delayed. Can you see it now?
Yeah, now we can see it. Now it advances. Perfect.
Okay, great. So I was just speaking about, of course, the entire landscape and how we specifically at our center, we're primarily interested in vaccines that will also serve for the global access perspective. This is just a slide to tell you a little bit of who we are. Even before coming to Houston 10 years ago, we were established as one of the product development partnership models that were even spearheaded by the Bill and Melinda Gates Foundation. Our niche has always been in the area of neglected tropical diseases, but we've also, as of 10 years ago, with actually our coincidence of moving down to Houston, we started a program supported by the NIH to advance a coronavirus vaccine program. This was done with the aspiration to collaborate. In fact, we collaborate with the University of Texas Medical Branch with a group of Dr. Ken Tseng, who, of course, as you know, he has a lot of experience in, the animal models for SARS, MERS, and now, of course, also COVID. And together with a group of virologists from the New York Blood Center, Dr. Shibo Zhang and Langying Du and Sarah Lustigman, in 2011, we started a program where we charged to take their know-how, their product, they were trying to advance a receptor binding domain for SARS and MERS, and we brought it into our facilities because we wanted to fashion it into compiling data that could really serve for two purposes. One, IND or regulatory enabling dossiers, while at the same time our niche, which is really the niche of process development, scale up, so that we could also technology transfer to manufacturing, whether it's pilot facilities or manufacturing facilities. In fact, our story with SARS and a little bit also with MERS is we managed to, in around 3 years, developed a process to, of course, scale up the RBD for SARS. We tech transfer the production process to Walter Reed. They actually manufactured a clinical lot, a drug substance lot. Unfortunately, we were, because of many different factors, as you know, lack of enthusiasm because the SARS was not as priority anymore. We couldn't really be able to move the SARS vaccine into the clinic, albeit we kept it, under regulatory and strict compliance for stability. And so we actually do have still our drug substance that it's quite suitable if we wanted to evaluate in the future. But as soon as we heard that we had, of course, this potential SARS-like virus, we resurrected our program. We, of course, looked at what we had done. We leveraged all the know-how that we had done between 2011 and, of course, 2020. And as you can see in the blue line, it took us less than 5-6 months to actually repurpose all our know-how and basically retook the gene of the COVID-19 and did exactly the same process for scaling and tech transfer and some of the preclinical development. And this is what I'm going to tell you right now. And just as a reminder, I think a few of these programs really highlight how the academic partnerships, and I think, the prior speakers clearly have highlighted this, that we are now a very important component in the ecosystem of accelerating, vaccine development in general. I think this pandemic has also shown how we can bring an enormous contribution to the field. In our case, we follow a very strict integrated product type of approach. We, in fact, fill the entire ecosystem from the discovery of molecules, doing, of course, the scale-up tech transfer, all the regulatory work, all the, preclinical work. We partner with the clinical trial sites. I think we've heard from the prior speakers a very similar approach, except that, of course, we are doing this in the nonprofit sector. So what we usually do in our labs, as I explained, we take, of course, either our own discovery of a target candidate or somebody else's discovery of a candidate. We fashion the product, the prototype of the vaccine. We look, of course, at the process, the yield, the purity, the stability. We develop assays that are for IND enabling or regulatory enabling release or stability. We also dabble a lot in formulation science, whether it's a buffer formulation or even adjuvant formulation. And depending on the program, we either partner or we do our own preclinical assessments. with specifically as relates to the SARS-2, the COVID-19 vaccine. We really framed our development on our lessons learned from SARS. Our capacity is in yeast expression technology, very similar, for example, to the technology used for the hepatitis B vaccine. We do this because we believe that novel technologies are great, but we always aspire to ensure that we also fill that potential gap of building capacity overseas and certainly transferring our technologies. We work a lot with the developing country vaccine manufacturers, and for them it's of course very easy to adopt technologies that they already have a know-how. So if you want to hear a little bit more about what we did for SARS, I mean, we have plenty of the publications. And now we repurpose all this. We did the same approach. We, for example, looked at the sequence. This is an example of some of the genetic engineering that we do. We know that yeast, of course, hyperglycosylates. We have to be cautious, of course, with the oligomerization of the proteins on the basis, of course, of their cysteines. So in this case, I show here how we took the sequence of the receptor binding domain, whether it's wild type. We then did a modification by deleting the first end glycosylation site. Then we did a second modification, which was a mutation in one of the free cysteines. And as you can see, as we manipulate and we look at how well we can control the process, we can reproduce, and we can certainly ensure that we have a good way of purifying our molecules. It shows that, for example, the molecule that we call the N1C1 is probably the one that is most appropriate. We even, of course, then do a lot of process development and scale up. Even without much optimization, we managed to reproduce with a very modest process. With very modest resins. We basically have a process that we could even get, without even optimization, around 300 milligrams of RBD per liter of fermentation supernatant. And of course, we now use this molecule to try to evaluate some of its functionality in our hands. We do also have a lot of capacity in the area of, I skipped one slide, there you go, biophysical evaluation, with not only ourselves, with cores around the Texas Medical Center. We look at biophysical comparisons such as dynamic light scattering. And it's quite interesting because even with the modifications that we do, they don't really affect specifically the area within the receptor binding domain that we know, of course, is going to be very important for its immunogenicity. But we do keep an eye, of course, on how the different constructs or the proteins coming from the different constructs, if we can keep monomeric, if we can see oligomerization, if we can see, of course, any kind of differences biophysically. Of course, what you all want to hear is what happens with move the next slide, there you go. The binding capacity. So we do two types of studies. We look at the protein on its own and how its ability to bind to a human ACE2 receptor. And as you can see, when we compare even wild type with the N1C1, they look very similar. We also look after we bind the protein on aluminum. Most of our formulations that we try to advance are alum systems. So usually it's either alum alone or alum in combination with other immunostimulants. In this case, I just show you data with alum alone. As you can see, we also titrate the ratios to see, of course, not only binding capacity, but also immunogenicity, and in this case, even binding to the human ACE2 receptor. And you can see that there's clearly no difference, even by reducing the ratio between the protein on aluminum. However, when, of course, we go to the in vivo studies, we do very simple mouse immunogenicity studies. On the left side of the panel, you see the total IgG. We actually can induce, of course, quite high in levels of antibodies. You don't see really a difference up to when you have greater than 1:4 ratios of the protein bound to the aluminum. So they're very comparable. It's also very comparable even when you use the wild type compared to our N1C1 molecule. And when you look at neutralizing antibodies, and we do this, of course, using pseudovirus assays, again, we also see that even with ratios of higher than 1:4, we can get some pretty prominent levels of neutralizing antibodies. Now, I know everybody would like to see what happens in a real challenge, and I don't have data, but I have to tell you that we just completed a study, of course, an immunization challenge study in partnership with Emory, with Dr. Sudhir Kasturi. He is compiling all the data right now, and we believe that this will be published probably after Thanksgiving. The data looks very promising, very aligned with all the other studies that have occurred in this model, even though I can already alert you that we do see some very, very nice neutralizing titers and, of course, protection also. So the other interesting aspect of this, I think I alerted, with my vignette, is as we were doing the process development and scale up, we were approached by Biological E. As you know, maybe all of you know, Biological E is one of the most prominent vaccine manufacturers in India. They do have a lot of experience with yeast-based vaccines, of course, including hepatitis B. And back in July, we provided them with a non-exclusive license. We basically technology transferred our research cell banks with all the know-how of our process. They very rapidly, I have to say, scaled it up. I think there's been news that they've already were able to scale up the drug substance up to 1.2 billion doses per year. We just got announced that the DCGI in India already gave them approval, and I believe tomorrow we're going to have the formal press release because the phase 1 / phase 2 trial just started, I believe, yesterday. They've already immunized from the first cohort of participants. So we're very, very excited about this partnership with Biological E. As you know, our vaccine center advocates for low cost, safe, affordable, accessible, adoptable vaccines. Biological E, of course, also is very much involved in the COVAX facility and is working together with CEPI and WHO and Gates Foundation. And so we are very hopeful that this may be one of the vaccines that can certainly come and fill a gap in, of course, the low middle income countries, which we're very, very proud of. I cannot finish my talk by saying that I'm the one speaking to you all, but at least from our group of core leaders, we have been working 24/7 since January pretty much. So these are just some of the few faces for you to look at that have been really behind the scenes doing most of this work. It's been quite intense, and I think we're all very proud of what we have accomplished, and we're looking forward to seeing how these new advancements will follow. And I will also, of course, end by thanking a huge amount of groups of funders. Our portfolio is quite an interesting portfolio. We put together funding from, of course, very generous support from the NIH, but at the same time, a huge amount of contribution by local foundations, international foundations, as well as, of course, US-based foundations. Our partners, not only from SARS-1 program, but SARS-2, PATH has been an enormous support, giving us a lot of technical advice. Groups like Millipore Sigma, who helps us with the scale up and process development, IDRI with formulation science, Emory, as you know, that I mentioned for our non-human primate studies, and of course, Biological E. So I would like to stop there, stop sharing and sending it back to you, Dr. Shankar, to see if there were any questions.
Dr. Bottazzi, it's fantastic. Thank you so much for telling us about the efforts that are going on Perhaps while we're waiting for questions to show up, I'll ask you a question. In going through your presentation, you talked about the IgG titers in the model systems. Do you know a time profile for these IgG titers? I don't recall seeing a temporal profile of how it changes.
Yeah, so when we do very simple immunogenicity studies, we usually do either 2 immunizations or 3. We want to look at the entire, capacity of, because in our hands, we primarily only have access to aluminum formulations. It's been a little bit hard for us to get proprietary immunostimulants. So we generally just look at alum. And so we do immunizations 21 days apart, or 1 or 2, and the data that I believe I showed in the slide was after the second immunization. So trying to align ourselves with that, everybody's trying to look at least at 2 doses versus 3 doses. We do know from the primate study that will be published soon, that was done comparing alum alone and a group with an alum plus. I know Biological E also will be advancing their vaccine as an alum plus. And again, I think that will dose spare and will also improve the immunogenicity. We don't know a lot about duration, as you know. I think not a lot of people know a lot about duration. We also do have some evaluation in T-cell immunology. Of course, as you know, we want to make sure that we have a balanced immune response. Alum is very Th2 driven, as you know. So I think that alum alone is giving us some very good responses, but I believe the intent is to make sure that it's complemented with a second immunostimulant. And I think that's where most of the, scientific community is supporting. But I think alum, and I believe Dr. Graham mentioned a little bit, and I know he's been part of a lot of advisories. Alum at the beginning was a little bit left outside, and I think now we're starting to see that it's getting some push, in including them, because I think if we want to have a vaccine that is of global access, albeit there's some wonderful adjuvant systems out there, it would really be beneficial to bring probably one of the safest adjuvants we've seen, and the fact that it's also quite available and affordable. So we're really, in our hands, we're really looking to see how we can reduce a lot the cost, increase a lot the access, and certainly make this quite scalable and accessible.
I mean, you almost answered my question. Because of the use of alum, you have Th2. I mean, did you monitor the Th1, Th2 ratio as a consequence downstream? And I didn't see that in the slide. So is that something that you did?
Yes, we did. And in the 2 preprint publications that we just uploaded that focus on, again, alum alone studies that we've done, we have, of course, shown that, it's as typical Th1, Th2, it's mostly skewed to Th2. We are waiting, as I mentioned, there's going to be probably the data from Biological E trickling in. And as soon as of course, we have that publication or even some non-confidential information, they will gladly share. We also, of course, have the data coming from the non-human primate also that we will be disclosing shortly. Yes.
Thank you. We still have more time for a couple more questions. So from the audience, Mark Oldham asked a question: what causes viral differentiation into relatively stable serotypes, as with dengue? Is it possible to predict whether COVID or other viruses might also differentiate into serotypes?
Mm-hmm. Maybe I'm not the most adequate to respond this, but I will try. I think, at least from our perspective, we are, of course, monitoring very closely how the virus is changing and modifying. The good news, at least that I know, is even within the RBD specifically, most of the changes are not occurring in what we call the receptor binding motif, meaning the one that is really the epitopes that are exposed. So right now we haven't seen, even by using, for example, different types of pseudoviruses, by putting different sequences in the pseudoviruses, we have not seen any differences in how certainly the response is induced, at least by our vaccine. But I believe that probably Dr. Graham can explain maybe better on what else should we be paying attention, in fact, if the virus is going to eventually evolutionary change. I think it's not changing very rapidly, as I'm understanding. Yeah.
That's the understanding I think people have in general, yeah. While Bali is loading his slides, there's one more question from the audience I'll post. He says, can you define binding capacity? That's a question from an audience. I'm assuming, yeah.
And I believe you probably, of course, mentioned how the protein binds to the hydrogel or the aluminum hydroxide. And in fact, it's binding 100%. So even at the level of below 1 to 4, we start seeing that it's not 100%, but anything of a 1 to 4 ratio, we definitely have an 100% binding capacity. And I'm more than happy to share that information with you separately if you send me an email. And I believe there's some data already in one of those preprint articles that we just uploaded. And we're slowly putting up all the data in these servers while we're, of course, waiting for the peer review evaluation of our work.
Fantastic. Thank you so much, Maria. I mean, it's spectacular talk. I mean, as always, thank you for your presentation. Now I'm going to have a great pleasure in welcoming Bali Pulendran. I mean, this is such a pleasure for me for multiple reasons. Number one, first, let me introduce him. Bali is the Violette Horton Professor at Stanford University School of Medicine. He's one of the pioneers in systems vaccinology. I've known him for years. He's got more ideas in 1 second than most people can think about in years. And he's absolutely phenomenal. He got his undergraduate from UK and then moved on to Australia to work with NOSAL for his PhD. Then came to US. He was in Emory for a long time, and one of the, young Turks when he was in Emory. Phenomenal scientist. And then Emory's loss was Stanford's gain. In the last few years ago, he moved to Stanford. Bali is well known for a phenomenal number of things. He's one of the pioneers in systems vaccinology, worked on several vaccines, flu and other things. He has now focused his attention on COVID, and I have no doubt that Bali will have enormous impact in the development of vaccines and the efficacy of vaccines in COVID research. I also have the pleasure of having Bali as a good collaborator and friend. So Bali, really welcome to this meeting. Thank you for joining, and it's really look forward to your presentation.
Thank you very much, Shankar, and good morning, everyone. I'd like to thank you, Shankar, and the organizers for asking me to talk in this very exciting forum at this meeting. So let me see. I would like to talk about some work that we've been doing to look at the immune response to vaccines and infections in the context of COVID-2. And mostly I'd like to tell you about work we're doing to understand the host immune response to this infection. And at the end, time permitting, I'd like to make a few comments on how I believe the systems immunology approach could be used to monitor immune responses to SARS-CoV-2 vaccines and to accelerate the testing of vaccines, and not just SARS-CoV-2 vaccines, but other vaccines that we might have to make in warp speed against other pandemics that might emerge in the future. All right, so let me spend some time telling you about our own journey when it comes to understanding the host immune response to SARS-CoV-2. I call this warp speed immunology quite facetiously. And our journey began back in, really it began in January, like for all of us. And I remember this very well. I was in a car in the parking lot of some grocery store. My wife and daughter had gone in to get something, and I was listening to the radio on the car, to the BBC World Service. And they were talking about these cases of viral infection emerging out of China. And I was thinking this could become a pandemic. So, we're an immunology lab. We work on understanding the immune system, a little bit on vaccines and infections, but obviously one wants to keep focused. And so it wasn't immediately clear that we necessarily wanted to look at this virus. After all, there were only a few cases emerging at that time. But I did contact all my clinician friends, the infectious disease people, one of whom is shown here, Professor Nadine Rupael, who is an amazing infectious disease scientist and a professor at Emory University. I called her up and I said, are you seeing any cases in Georgia, Atlanta? At that time in the Bay Area, we were not seeing very many cases at all. And she said, yes, we are, and they seem to be on the ascendancy. And then I contacted some of my international friends, one of whom is shown here, Professor Malik Pires at Hong Kong University. As many of you may know, Professor Malik Pires is a world-renowned virologist. He was the man who isolated the SARS virus initially years ago when the first SARS-CoV-1 emerged. And so he said, absolutely, they've been seeing cases in Hong Kong, many severe cases. And I said, right, we need to work on this. So I contacted our Stanford Biosafety office on March 3rd and told them, look, we want to get PBMCs from these infected people. And I have to say that they were extremely helpful and cooperative. And so by the end of the month, we had the biosafety permit. We had approval, the import permit approval from the CDC. And April 10th, samples arrived by World Courier. And 3 weeks later, we submitted the paper to Science. And I think one of the reviewers must have gone off on an extended summer holiday, because then we only got the comments back to our revision short before then. The paper was accepted and then published on August 11th. So I want to tell you about the data we've generated in that, go through that, and hopefully it'll reveal some interesting concepts about this, about the immunology of this disease, but also to tell you a little bit about what we're doing now in the future. And so when I say we, it's not me at all. The people who did the work, it's an outstanding young postdoc, Prabhu Arunachalam, together with two other outstanding postdocs, Thomas Hagen and Florian Wimmers, with a fantastic MD PhD student, Madeline Scott, working in the laboratory of Purvesh Khatri, and Holden Maker, the head of the Immune Monitoring Corps at Stanford. So this was our sort of SARS-CoV task force one. We have many task forces, but this was one task force. And so that was the paper. So what did we do? Well, we wanted to analyze the host immune response to these humans who were infected. Of course, as you know, infection results in mild or moderate disease, severe disease, or critically ill disease. And we were able to obtain blood samples from these patients, and we applied the tools of systems immunology that are shown here. This machine, CyTOF, can measure, it's a mass cytometry machine that can measure the cellular composition. You can use 40 different antibodies, RNA sequencing, O-link, metabolome, neutralizing antibody responses. So our questions were quite straightforward, and it was a question that all immunologists were interested to ask. What is the nature of the immune response to COVID-19? Is there an immune signature that can predict disease severity? And what is the immune response in asymptomatic individuals? I'm not going to talk about this today, but we are looking at asymptomatic individuals and seeing some really interesting things. So let me tell you a little bit about our CyTOF analysis. We got 40 samples from Atlanta, 100 samples from Hong Kong. Now remember, shipment of samples across internationally, the CDC import permit required that these had to be fixed. So these PBMCs were fixed in 2% paraformaldehyde and then shipped. But luckily we have a CyTOF panel that can work even with fixed cells. So what did we find? This is a Tisney plot showing all the different types of cells in human PBMCs, you know, B cells, basophils, T cells, and so on. And a striking finding was that the frequency of plasmablasts. Plasmablasts are cells that produce antibody. These are called antibody-producing cells. Massive increase in plasmablasts and massive increase in the frequency effector CD8 T cells. Effector CD8 T cells are T cells that are highly activated and that produce cytokines and that can kill virally infected cells. So clearly there was a very strong adaptive immune response going on in the cohort in Atlanta. Also, similar findings in Hong Kong. And this was important because if you remember, back in the early days, as the pandemic was emerging in different parts of the world, a question that we all had was, would the disease manifestation and the immune response be similar in these patients in different parts of the world, or would there be differences? And so we were quite keen to compare these two different cohorts. And again, we see very similar effects in the plasmablasts and CD8 T cells. Another interesting thing was that the frequencies of plasmacytoid dendritic cells. Plasmacytoid dendritic cells are also called PDCs, are cells which are very rare in the body, less than 0.5% of all cells in your blood. But their rarity doesn't diminish their importance. They are probably one of the most important cells when you think of viral infections. Why do I say that? I say that because they are the cells that are highly adapted to produce interferon alpha. And interferon alpha, as you know, is one of the most important cytokines in controlling viruses. And what we saw was that in these SARS-CoV-2 infected patients, the frequency of plasmacytoid dendritic cells was diminished. Also, when we looked at the intracellular staining of this expression of PS6. Now, what is PS6? This is a molecule that is downstream of mammalian target of rapamycin, which, as you know, is a critical metabolic regulator in the mammalian immune system. And what we found was that there was diminished expression of PS6 in these plasmacytoid dendritic cells. Now, you might think, well, so what? I mean, that's just a marker. But as it happens, we had shown previously in a paper that we published several years ago that this molecule, PS6, shown here, was acting, of course, we know it was acting downstream of mTOR, but it was really important in mediating interferon alpha beta production in plasmacytoid dendritic cells through this IRF7 transcription factor. Wow, so we thought, so does that mean that in these SARS-CoV-2 infected patients, the plasmacytoid dendritic cells were unable to produce interferons? So we did the experiment. We got the PBMCs, and we stimulated the PBMCs in tissue culture with these viral cocktail. What do I mean by viral cocktail? Viral cocktail is a cocktail of TLR ligands that resemble a viral infection. So TLR7, 8, TLR3. And what we saw was that if you stimulate those PBMCs from a healthy person, shown here, and you measure by intracellular cytokine staining, interferon alpha versus TNF alpha, you see lots of production of interferon alpha and TNF alpha. But look what happens in a SARS-CoV-2 infected patient. In fact, there's a greatly diminished response. And this is not just one example, because you can see this is a very robust finding. By the way, I have to tell you, if you don't stimulate the cells in tissue culture without any stimulation, there's no production of any of these cytokines. Okay, so what about, that's interferon alpha, but what about pro-inflammatory cytokines like IL-12 and IL-6, TNF alpha? Well, let's do the experiment. Let's get PBMCs from healthy people or infected people and just culture them without any stimulation, or with bacterial TLR ligands, which is lipopolysaccharide or flagellin or PAM3-cis. You see a very nice production of IL-6 and TNF alpha in these healthy people, but very surprisingly, we see hardly any production in the infected people. Now, this is looking at myeloid dendritic cells, okay, but the same thing applies to monocytes. What about if you use viral TLR ligands? Again, very nice production in healthy people, but much less in people infected with SARS. Now, you can see the stats there. It's a very significant finding. So this was odd because, you know, as we were writing up this paper, we were seeing all these papers coming out in preprint archives saying, oh, in SARS-CoV-2, in the plasma, there's lots and lots of IL-6, there's pro-inflammatory cytokines. And yet, when we did this experiment, when we took cells from the blood and asked, well, can those cells be stimulated to produce these cytokines? We didn't see any, or we saw greatly attenuated responses. How can that be? That seems so paradoxical. So we said, well, let's take a look at the plasma or serum and see if we can find any of these pro-inflammatory cytokines. And indeed, when we did this, and we did this using Olink, and you can see IL-6, TNF, MCP3, you know, ICU, those are critically ill patients, severe patients, moderate patients, healthy patients, you see. So there is this enhanced production of these pro-inflammatory cytokines, entirely consistent with what was being published in the literature. And so how can that be? How do you reconcile this finding with our earlier finding that there was no reduced capacity of the cells to produce? So to get a deeper insight into this, well, before I show you that, I must tell you 3 new cytokines. When I say new, what hadn't been reported in the literature that we observed in our study was oncostatin M, TNF superfamily 14, and this molecule called NRAGE. These are very interesting because, as I say, they hadn't been reported to be increased in SARS-CoV-2 patients. However, these are well-known molecules that are involved in inflammation, and in fact, these have been implicated in mediating inflammatory processes in the lung in response to other kinds of lung inflammatory disorders like COPD. And so we were very interested, and we're pursuing these molecules as potential therapeutic targets. We're working with Biogen, for example, who have reagents who can block this pathway. And so those experiments are ongoing. All right, but to get back to this paradox that seemed to exist of why these PBMCs are unable to respond and produce these cytokines, but yet we see copious quantities of these cytokines in the plasma. What we did was this technique called CytSeq. Many of you know what CytSeq is. Basically, it's looking at the single cell transcriptomics of cells in the blood. But what we can do is also use antibodies to stain for surface markers of these cells. So in this way, you know which cell types we're looking at, because each antibody is linked to a nucleotide sequence which acts as a barcode that then you could use primers to amplify, so you can define the population of interest. So we did CytSeq analysis on 8 SARS-CoV-2 subjects, 9, 3 severe people plus 1 ICU patient, 3 moderate people, 2 convalescent, 5 healthy, and then 2 people infected with influenza A and 2 people with RSV. So we had frozen PBMCs, and what we did was to enrich for these very rare population of cells that I mentioned, like plasmacytoid dendritic cells or myeloid dendritic cells. Why did we enrich for them? Because they're rare, and we want to make sure that they are well represented in the final population of cells we're looking at. So we enrich for them and then add them back into this mixture of total PBMCs in a 1:2 ratio. So they go from being less than 0.5% to now suddenly like 20 or 30%, okay? So what do we see? Well, here's a UMAP cluster, and it's very pretty. You can see all the major cell types here, including hematopoietic stem cells, NK cells, RBCs, and so on. You even see contaminating red blood cells here. Well, if you now ask at a very broad level, what is the transcriptional landscape of COVID-19 patients relative to flu, healthy, or RSV? You can see there's transcriptional activity is evident in most of these cell clusters. And there are some unique clusters here. For example, flu has this cluster here. RSV has a cluster here that doesn't seem to happen with SARS-CoV-2. Now, if you then say, okay, we have convalescent people in green, purple, severe in orange, how do they compare? Now we can begin to define transcriptional spaces in these clusters that seem quite distinctive between these different disease states. So the orange seems to have unique areas where the purple doesn't occur. And then the green convalescent area is again occupying quite a different space that seems to overlap quite a bit with the gray space shown here in healthy. So by 2 months, when I say convalescence, these are 2 months after recovery. I think people seem to have recovered their transcriptional state to what you would see in a healthy person, more or less. But let's get back. Let's dive into this data a little bit more. Let's talk about interferons. I told you that we couldn't get these cells to make interferons. So the obvious question was, could you find the messenger RNA for interferon alpha and interferon beta in this data set? And there you go. You see a vast sea of gray and not a single red dot here and here. So there's no message for interferon alpha and interferon beta in these PBMCs. Oddly enough, when we looked at other genes like IFI27 and IFIT-M3, these are genes that one calls ISGs, or interferon stimulated genes, or IRGs, interferon response genes. What we saw was that you can see that the expression of these ISGs were present in many of these clusters. So this was a paradox. If these are interferon stimulated genes, and if the cells are not making interferon alpha and beta, how on earth are we finding these transcripts? So we were quite worried. We didn't know how to interpret this. So we said, listen, let's take a broader look at all these ISGs, not just these two molecules I've shown you in the previous slide, but these are a list of all the known ISGs. We went to the literature and we said, how many ISGs are there? Can we go one by one and see which ones are being expressed in these PBMCs? And what we can see, as you can see, there's quite a few of these ISGs being expressed in these patients here. Who are these patients? COV5, COV6, and 7. Let's see now, those are the moderate people. Oh, that's interesting. Now, as you go further down, see the red, not much. Maybe there's a cluster here. And then this red, not much. And then this, who are those red people? Oh, those are the severe people. And then the green, those are the convalescent people, not much. And then healthy are these people shown in black, not much. So at first we thought, wow, there seems to be an association of the expression of these ISGs with disease severity. Now that's just looking at this CDC2 cluster, which is the classical dendritic cell 2 cluster. What about the plasmacytoid dendritic cells? Again, a similar sort of pattern. But you know, this was only with 9 SARS-CoV-2 patients, but we wanted to be sure that this result was reflecting what was really happening. So what we did was say, let's take a look at bulk RNA-seq in many more SARS-CoV-2 patients, and I think we had 30 here. So what we did was to analyze the expression of these ISGs using bulk RNA-seq, and then what we saw was, yes, there was high expression of these ISGs in some of the patients. Again, you can see that there's a moderate patient here, but these are 2 severe patients here. Here's a moderate patient here. All right, so that correlation with disease severity was there, but not so clear with the bulk RNA-seq. But what was much clearer was the correlation with the days post-symptom onset. You see here, that's the green scale here. So lighter green is very early after disease symptom onset, 5 days, 10 days. You see, it's expressed at high levels there. As you progress down towards later stage of disease, you see this is much less evident. And then if you do a covariate analysis and ask, what are the covariates that could explain this ISG? Age, not really. Disease severity, perhaps. Time post-symptom onset is a major variable. Gender, not really. And then there's all these residual factors that we don't know what they are. We're looking into that. We don't yet know what they are. So we said, okay, it seems that there's a time dependency of these ISGs, which are genes switched on by the interferon response. But where is the interferon coming from? We haven't seen it. So we measured by Luminex and we couldn't find any. At that time, we came across this exciting new technology called single molecule array detection, the SIMOA technique that was being developed by this company. And so we worked with this company to measure interferon alpha. And I can tell you the sensitivity of this technique is so high that you can measure femtogram per ml quantity. Remember, 1,000 femtogram is 1 picogram, okay? Your ELISA kit, the sensitivity is about 10 picograms per ml or so. So this is really very sensitive. And what we saw is this beautiful time dependency. You see, days post-symptom onset, at around 10 days or so, there seems to be a peak, but then there's a rapid decline. And now we have many more patients that really have confirmed this. So there's an incredible time dependency. So it's coming early on and it's switching it on. But where is it coming from? Because I can't see it. We can't see it in the PBMCs. So we analyzed the concentration of interferon alpha that we measured by SIMOA technique on the x-axis versus the ISG score, which is the genes that were stimulated by interferon alpha, and you see this beautiful positive correlation. So the model we have is that SARS-CoV-2 is switching on this transient early interferon alpha that is then turning on these ISGs. Now, a week or two before we published our paper, our colleagues at the Pasteur Institute came up with a similar result showing that the ISG score was impaired in SARS-CoV-2 patients. In their finding also, they see this dependency on disease severity. They didn't look at the kinetics, but there does seem to be a dependence on disease severity. Same thing with interferon alpha. They also used a similar technique, getting quite similar results to what we were getting. Subsequent to the publication of our paper, maybe a month or so later, the group of Adrian Hayday published his paper in Nature Medicine, showing again with interferon alpha, again measuring in the serum. You see there's this time dependency. Early on after infection, there's high levels. Again, this is femtograms per mil. You see the levels are quite low, but as the disease progresses, you find diminished response. And then there are other papers showing impairment of this interferon alpha. And this was the earliest paper from the group in New York University from Benjamin Tenover, showing that SARS-CoV-2 is inhibiting interferon alpha. But then there are also studies. For example, this excellent paper from the group of Yale, where they did a longitudinal analysis of patients, and this was the first longitudinal analysis to be done. There they see, in fact, severe patients seem to have higher quantities of interferon alpha. So I think what this is telling is that it's not so much the disease severity, but it's the kinetics that seems to matter, that all patients initially develop interferon alpha transiently, and then that goes away. What else did we find? We found in our site-seq data that the expression of HLA-DR, which is a class 2 MHC molecule, was reduced in classical dendritic cells and classical monocytes in these SARS-CoV-2 infected people, particularly in the people with severe disease. We went back to our CyTOF data and said, is that true? And indeed, that is true in both the Atlanta and the Hong Kong cohort, okay? And we also found that this gene called S100A12. Now that I had never heard of that gene before, and I suspect that many of you may not have heard of that, but it's very interesting because this is in fact this gene that encodes that molecule NRAGE that I mentioned to you before, that we saw by Olink. And I'll come to what NRAGE is doing in a minute, but this molecule was expressed in these monocyte clusters here. And we said, well, and what we saw was that there was this inverse correlation between the expression of HLA-DR and the expression of NRAGE. So in other words, in SARS-CoV-2 severe patients who had low levels of HLA-DR, they also expressed the highest levels of NRAGE. And if you do a correlation between the site-seq data shown here and the expression of the serum Olink protein levels, see a beautiful correlation. Now, why is this interesting? This is interesting because this phenotype of cells, myeloid cells that express low levels of HLA-DR and high levels of S100A12 or NRAGE, is the phenotype of cells, what you call myeloid-derived suppressor cells, MDSCs. So it was fascinating in these SARS-CoV-2 patients. While there was rampant inflammation going on, lots of IL-6 and TNF and all of that, we were seeing this innate refractoriness. Cells are unable to respond in vitro to TLR ligands, and the emergence of these myeloid-derived suppressor cells. How is this happening? How can an infection in the lung result in all these amazing changes that are happening so far away in the blood? Well, maybe the virus is coming into the blood. We tried very hard to detect virus in our RNA sequencing data, but we could find no viral copies in our data set. What else could it be? Well, maybe there's bacteria in the upper respiratory tract. We all have microbiome, and maybe that's playing a role. So we said, let's take a look at bacterial products like endotoxin or bacterial DNA. And what we saw was that in the severe patients or people critically ill, there's high quantities of lipopolysaccharide compared to healthy or moderate, and higher quantities of bacterial DNA. And interestingly, when we correlated these bacterial products shown here, bacterial DNA versus cytokines made like IL-6, TNF, MCP3, NRAGE, oncostatin M, TNF, superfamily 14, we see a beautiful correlation, arguing that there could be a role for these bacterial products in mediating this excessive inflammation in the lung. And so how is that working? That's something that we're looking at now, but I can tell you we have data that we're confirming that this is mediated by metabolites. So in other words, the bacterial products are triggering the production of certain metabolites that are having this suppressive effect in the periphery. So even whilst there's so much inflammation going on in the lung, and that's now been shown by others where they've looked at lung tissues and they see lots of IL-6 and TNF, there seems to be this feedback in toning down what's happening elsewhere. So this summarizes this. Basically, I think the data so far shows that early in disease, we're seeing low levels of inflammation, but this early low type 1 interferon that is turning on ISGs in these cells in the periphery. But as the disease progresses, there's excessive inflammation, production of inflammatory cytokines, bacterial products. And in fact, what you see is there's suppression of innate immunity in the periphery. This might seem paradoxical, not only to an immunologist, but to anyone. This seems quite schizophrenic of the immune system. You see, why is it responding with so much vigor, lots and lots of inflammation coming out of the tissue, but elsewhere, the innate system is sleeping in hibernation? Why? Why would evolution design a system like that? It's a teleological question which is impossible to answer, but we have speculation. And our speculation is that there's what we call spatial dichotomy in the immune system. In the mammalian immune system, there's a tissue infection, SARS-CoV-2, at least initially in the lung, where there's excessive inflammation going on, but there's suppression of innate immunity elsewhere. The spatial dichotomy, we think, is an evolutionary adaptation so that to prevent the host from developing systemic inflammation very early after a tissue infection. Imagine if these cells in the blood were also responding very vigorously and producing lots and lots of IL-6 and TNF and all these inflammatory cytokines. Probably the prognosis would not be very good, would it? Because, you know, rather than having a fatal end in 2 weeks or 3 weeks, the whole thing might be over in 2 days. So we think it's an adaptation. So it's a fundamental process. And as we read the literature, this has been seen in other diseases like sepsis. And the question is, what is the mechanistic underpinning of this? What is factor X that communicates between the lung and elsewhere? And factor X is what we're looking at, or many factors, many factor X's. That is the mechanistic angle that we're now pursuing with some really exciting data. So in the remaining 2 minutes, I want to conclude by saying, I did tell you in the beginning, this approach of systems immunology that I've sort of highlighted with this example of SARS-CoV-2 can also be applied to vaccines. And we and a number of other labs have been focused on doing this. So here's an example from a paper that we published a few years ago, where you applied high dimensional flow cytometry, Luminex analysis of cytokines, plasma metabolomics, blood transcriptomics, and then constructed a multi-scale, multifactorial response network that could predict the immune response to this shingles vaccine. We've applied that, and this was a collaboration, a fantastic collaboration we had with Shankar's lab. It seems so recent, but it was quite 5 years ago, where we said, can we apply systems approach to predict the durability of the antibody response to vaccines? Now, isn't that something that we would all be interested in? We've heard these amazing talks from our friends who are making vaccines, which is a fantastic thing that's happening. And we're all anxiously waiting to get those vaccines, but also we're all worried, are we going to be protected in 6 months from now? So we applied this to this seasonal flu vaccine study. As you know, seasonal flu vaccine is a good vaccine, but unfortunately it's not very durable. And you can see many of these people develop a hemagglutinin titer at 1 month, but then many of them fall below what you need for below a certain threshold. When this is an arbitrarily defined threshold, some of them do not. And so the question was, could we have predicted this? So we call this temporary and persistent responders. And the answer was, we did come up with a signature at day 7. And this was comprised of genes involved in platelet activation. You might think platelet activation and durability, what does it have to do with that? But why I'm mentioning this to you is now we validated the signature with other adjuvanted vaccine, including some adjuvants that are now being already in the clinic, licensed for use, and we've seen that this can predict durability. So we're very excited to work with COVID vaccine manufacturers to see whether we can apply this kind of systems approach to predicting vaccine outcome. I mean, I think it could be useful, perhaps less so for COVID, but more so as we think about post-COVID vaccinology. and which is really my last couple of slides. As you think about vaccinology, my good friend Rino Rappioli wrote this wonderful review where he divided vaccinology, 1980 vaccinology, 2010 vaccinology, and 2020 vaccinology. Vaccinology 1980 was highly effective. We've had so many amazing vaccines that have saved hundreds of millions of lives, but it was long and protracted and would take 10, 15 years. With the advancement of technology, including Reno's reverse vaccinology, neoadjuvants, this discovery process became shorter. And as I've mentioned to you, with the advent of innovative trial design and systems biology, we hope that this process can become even shorter. And what we have now, as you've heard from all our friends, is warp speed vaccines, where this is an absolutely remarkable and brilliant and amazing thing that's happened in things that would normally have taken 10, 15 years are taking 9 months. So my question to you, my friends, is as we now move forward into the, previously it was vaccinology BC, before Corona. Now it's vaccinology during the time of Corona. What is vaccinology AC after the time of Corona going to look like? Are we going to go back to this model? It seems, if so, that would be unfortunate, because that would mean we haven't learned anything from this warp speed. Or perhaps we're going to continue with warp speed. I don't think so, because that will bankrupt the world economy in 1 month. Because if every vaccine you say we're going to have 100 candidates we're going to test, that's not sustainable. I submit to you, my friends, that vaccinology AC, systems vaccinology, has an enormous role to play in the early monitoring of vaccine responses, in using these tools that I've told you about to predict vaccine responses, either in early phase 1 trials, you can have correlates of signatures that predict durability, or in retrospective nested case control studies in phase 3 trials, or if we have a challenge model in these controlled human infection model. So with that, let me thank the people who did the work. As I said, this was really an amazing team effort, including these people listed in my lab, and I already mentioned all the other people who we're working with. So with that, I'll conclude. Thank you.
Bali, it's absolutely stunning, phenomenal talk and a tour de force. Thank you very much. It's a great presentation. I'm going to take the prerogative of being the moderator to ask you a couple of questions. The first one deals with, you talked about type 1 interferon response and the temporal, kinetic, longitudinal behavior of that. Have you looked at the type 3 response, especially interferon lambda, where the responses are restricted to mucosal surfaces and they are really beneficial in various ways? So therefore, have you looked at the kinetics and as well as in blood, what happens to these interferons?
Yeah, great question, Shankar. We haven't, but it's clearly extremely important, because as you've said, that is the lambda is produced by mucosal tissues, and there are already phase 1 trials going on to test lambda 2 that I'm aware of. And so I think it's an important target, and we haven't, but we should look at that. Yeah.
The other question is, you referred to the 10-over paper. We have a very interesting paper. So they, also similar to you, showed about the interferon response genes, but they also talked about the inverse relationship with chemokines. Have you looked at the kinetics of the chemokine response in blood in following up on their, logic of chemokine correlation, anti-correlation?
Yes, we didn't in this initial paper, but as I mentioned to you, we are now doing the work with asymptomatic patients and looking more broadly at many more severe. And, is very fascinating. If you look at asymptomatic patients, what sets them apart is their unique chemokine profiles compared to people with moderate or severe infection. So we're still scratching our heads wondering what it all means, but I think the chemokine axis seems to come into play when you look at asymptomatic. And these are truly asymptomatic patients, because these are household contact studies that we were lucky to have access to samples from. So.
Oh, great. While Liang Schweitzer sets up her slides, I'm going to ask you one question from the audience. If you think, this is regarding your teleological comment, if you think this is an evolutionary adaptation, how does this relate to the way children have been responding to COVID?
Yeah, well, that's a terrific question. the neonatal and infant immune system is in a nascent state. It's maturing, and many of these mechanisms that we see, a little bit of refractoriness, innate refractoriness, myeloid-derived suppressor cells, might be a part of the ontogeny of the immune system in the first few months of life. That's just pure speculation. No one's really looked carefully, you see? And so, you know, that's something that we need to look at to see to what extent are we seeing these sorts of mechanisms coming into play in infection in children, you know, so.
Bali, thank you very much. I mean, amazing talk and very much in-depth. Thank you. I appreciate very much your really erudite talk. And now I would like to invite Dr. Liang Schweitzer. It's my pleasure to invite Dr. Schweitzer. She's the co-founder and president and CEO of HIF iBio emerging multinational biotherapeutics company. Prior to this, she was the co-founder of Harbor Biomed. She served as the head of the Asian Cancer Research at Sanofi before moving to her own enterprise, and she was the director of leaks evaluation at Bristol Myers Squibb prior to that. Dr. Schweitzer graduated from Science and Technology of China, University of Science and Technology of China, majoring in biology. She got a doctorate in microbial engineering at Minnesota. I'm sorry, she got her master's degree in Minnesota. She got a doctorate in University of Zurich, working in molecular biology. And then most importantly, she trained with the great Harold Warmus in Memorial Sloan Kettering for her postdoctoral research. Dr. Schweitzer, it's really a privilege to have you talk to us today, and we look forward to your talk. Welcome to this forum.
Thank you very much for the kind introduction. And I also want to take the opportunity to thank the meeting organizer for this 4-day symposium. It's really timely and a lot of wonderful talks, useful information. So I'm really glad to have the opportunity to tell you about the development of an effective neutralizing antibody to treat COVID-19 at Hi-Fi Bio here. As you heard, when we're facing those pandemic, we all wanted to come up with solutions. At Hi-Fi Bio, what we have established is outstanding expertise centered around drug discovery and development. As you know, I myself being in industry many years doing oncology drug discovery, immuno-oncology drug discovery, and also centered around immune modulation. Around at Hi-Fi Bio, we have had a lot of expertise how to identify antibody therapies. We also working open innovation model, working with top academic as well as industry partners, try to build a pipeline of sustainable therapies centered around immune modulation. So one of our leading expertise and technology is the single cell technology, where we could, using single cell analysis, looking at immune modulation, quickly identify immune repertoires, B cells, T cells. So when this pandemic break out, so we at Hi-Fi Bio thinking about, can we use what we have already established so far, try to come up with antibody solutions targeting COVID-19. So when we think about landscape at COVID-19, what we have see, you know, there's a lot of efforts with vaccines and also other antiviral efforts. One of the idea we had early on in February said, what happens if we get convalescent serum from the patients and looking at B cells also from PBMCs, what we can identify potential antibody from the patients themselves. So we then reach out, as previous speaker mentioned, to different hospitals, academic institutions, try to see whether we can establish the collaboration with them, identify antibody therapies. So by May, we're working with multiple hospital and academic institutions. We selected innate antibody and start thinking about how to optimize those into drugs. So we start thinking about drug desired drug properties for targeting COVID-19 spike protein. And we then also start thinking about manufacturing. For any drugs getting to patients, manufacturing normally takes about 14 to 18 months as a standard process. And thinking about in this pandemic, we try to figure out, is there a way to get rapid process to bring the drug to patients? So we actually managed to squeeze it within 3 months. So once again, resonate with Bali was thinking about the vaccine development. Antibody development also went into unprecedented speed. So from start working on that to IND filing 6 months, I've had many years in industry file more than 20 plus INDs. Never in history we have had achieved that speed. And after we filed IND, we started first in human. Right now, we are having the first cohort completed and starting the process of completion for the second cohort. So once again, we were looking forward. When can we get potential indications for filing? So we're aiming for the first quarter of next year. So what is the ideal neutralizing antibody to help the patients? What you have to think about, first of all, is safety. We wanted to make sure that anybody had no serious side effects. We also wanted to be effective at reducing the viral load. And more further on, thinking about future, can we supplement with vaccine approaches for a prophylactic setting that is suitable for all patients? So we named our antibody HFB 3013-2A. This antibody, when we design the engineering to make it into a drug-like molecule, we thought about multiple aspects on all 3 fronts. So first of all, to be safe, one of the things people encounter early on in dengue virus and later on SARS-MERS is thinking about antibody-dependent enhancement, ADE for short. So we wanted to engineer our antibody to minimize the ADE risk. We also want to minimize inflammation risk. And since this is a fully human antibody, we should have a very low immunogenicity risk. That's another aspect of making antibody drugs people have to think about. And also when you make a drug, you also have to think about a distribution in patients. So if we can make this drug distribute into the lower and upper respiratory system, that would be helping for the fighting viral infection. And as well as when you get antibody in people, although compared with small molecular antibody already have an advantage of being longer half-life, is there a way to extend this half-life further so you don't have to keep on going to get additional boosts of doses? So that's one thing we also have considered seriously. And finally, you wanted to be able to, for prophylactic setting, ideally it will be easy to administrating. You wanted to have outpatient method to do that, and you also want this antibody can be easy manufactured. So we're very proud to say that we have achieved on all those fronts. First of all, this antibody is a fully human IgG4, so we have no immunogenicity, and we have reduced ADE risk. I'll show you the data in a minute. And we could complete neutralizing, and based on our structure analysis, we actually have covered majority of the spike and ACE interaction. And we're also looking at this antibody with different mutation settings, including early on the prominent D614G. And we're thinking about how to improve distribution, at least in preclinical model, and now we're connecting clinical evidences. Well, this antibody is distributed, favorably in mucous membrane and lung. And we engineered this antibody have extended half-life. So we can have, through single injection, we predict up to 12 months based on the preclinical model, as well as we just initially obtained some clinical evidence as well. So this antibody we made is very easy to produce, gettable, and we are doing intramuscular as subcutaneous formulation. So hopefully we can give injection, one-time injection to patients, achieve desired outcome. So here is a summary of many months of work. Initially, we wanted to have an antibody that binds to the spike protein very well. So we achieved sub-nanomolar binding. And we also looking at blocking of the interaction that you can see that we also have nanomolar range activity. We also then subsequently tested pseudovirus entry as well as live virus entry assay. And once again, we can achieve desired efficacy. And because of this engineering, and you can see in non-human primate preclinical evidence, we have 10.5, 7.10 days that projected clinical half-lives up to 78 days. So with that in mind, we then designed in vivo efficacy data. So with 2 different doses, 10 mpk and 50 mpk, we demonstrate effective antiviral efficacy. And we also, in this efficacy model, we looked at the viral clearance in the lower respiratory, upper respiratory system, and it comes to desired outcome as we hope we could achieve. So looking at this antibody, and we collaborated with academic structure experts to do crystallization on this together with the spike protein. And as you see, the spike protein and ACE2 interface have 17 amino acids. That's the dark red plus the blue here. So our antibody, which is the red and the pink surrounded around it, it actually interacts with 29 amino acids with the spike protein. Only 2 amino acids here does not cover where the ACE bind to spike, but entire 15 out of 17 was covered by this antibody. And we subsequently did modeling centered around all those mutations that we have been identified. And as we're going through every day, new literature, new mutation comes up, we do modeling. And based on our interactions surface, as well as the location of the model, we predict that our antibody would working on the mutant as well. And indeed, that was the case. As far as we can access those mutant reagents, as well as the neutralizing acid that is provided, we demonstrate this antibody have good binding, not only to wild type, but to mutants tested as well. And we see good effective neutralization of this different mutations. So one of the things we thinking about is engineering to reduce the ADE risk. So compare with this, this drug is IgG format, as IgG4 format, as I mentioned. Compare with its IgG1 version, you can see the reduction of different Fc gamma receptors. And this subsequently translated into an in vitro ADE induction assay that we have compared with positive and negative control here. So we think this antibody have been generated in an engineering way to have reduced or minimum amount of AD activity, and also associated exacerbation of inflammation due to Fc gamma binding. So with the engineering to extended half-life I mentioned in the summary slides, and here is show detailed data, and you can see the antibody amount consistently exists in the cynomolgus monkeys when we performed the PK analysis. And this is achieved by enhanced FCR and binding, which helps the antibodies getting a prolonged half-life and up to 78 days based on our modeling. And we also know through this, we can also enhance the delivery to the mucosal tissues. So all those data have now been just initially validated from the first cohort PK study. We have observed results, and so far the data we observed predict really well, and with the modeling we did based on the non-human primate study. So everything is as we have engineered, demonstrate the extended half-life in human. So to ensure, we have safe doses when we get moving to clinical trial design, we then, performed the tox studies, and we have had 50 mpk and 300 mpk in non-human primates, and with 2 different doses and 1 week apart. So far, we can have the novel amount of 300 mpk and no marked accumulation and minimum ADA from our non-human primate study. And we also looking at multiple readouts through the study. For example, we don't see any changes in body weight, the quantitative food consumption, body temperature, all the other changes you could imagine in the GLP tox setting. We don't have any findings that would really make us have any safety concerns so far. So with that, we then went into phase 1 healthy volunteer study. The idea of the phase 1 rationale is to ensure we have a safe, tolerable, durable PK over time, and evaluate extended half-life. So we looking at the exposure of the drugs over a period of time up to 270 days for the safety follow-up. And we also looking at the biodistribution in mucous membrane. So so far, right now we are ongoing with the phase 1 study. Cohort 2 are being actually dosed already. Just based on the news I heard this morning, everyone is doing well. And the 3 doses we have is 400 milligram, 1,000 milligram, 2,000 milligram, based on our efficacy study in non-human primate. And based on the results we got so far from 400 milligram, we are confident those are the 3 dose ranges that will really have strong efficacy, cover variety range of viral node. And we will start to proceed with phase 2/3 after day 30 primary readout. We don't have to wait until 270-day safety follow-up. FDA is fine with the data we're getting for 30 days to proceed for phase 2/3. So the thinking centered around phase 2/3 is as follows, that we have thinking about, what are the most effective populations we're going to position this drug into, and looking at the data accumulated so far, also learning from other antibody clinical trials. We noticed that 70% of hospitalized patients have underlying risk factors, and out of that, 94% of the deaths are the patients with underlying risk factors. So clearly the patients have 1 or more risk factors, such as age, non-disease, existing heart conditions, obesity, diabetes, and those are the population have a high risk to progress to severe diseases. So we will design our clinical phase 2/3 trials in multiple countries, and looking at reduction of viral load as well as prevention of serious complications. So those are the currently the feasibility of phase 2/3 are ongoing right now. We're looking at potential countries and patients where we can enroll, and we plan to start this trial as soon as we got 30-day results from the phase 1. One of the things we talked about is also this antibody is well positioned for prophylactic setting, because we know it can be injected using intramuscular injection, for example, and it can clearly, effectively to reduce the viral load, even preventing virus from infection if you already administered it prior to infection. So we could thinking about the population where they don't get vaccinated for one reason or the other, whether it's health conditions or other reason they don't have access to vaccine, as well as those healthcare workers populations that have close contact, high exposure to the viral infection. So you could think about, in addition to vaccine, you give them first line of defense of this antibody. So we're considering and planning a prophylactic trial plan, and where we definitely need more patients to complete the trial. And the fact that this antibody can persist in the body up to 270 days or up to a year, depends on the dose, your administration. That could really provide a good protection for all the people getting this antibody against viral infection. So thinking about, the earlier talks talking about, we see, potential for other coronavirus or even other infections. How do we, at High-Five Bio, where we have rapid capabilities to respond to infection, which we, through our single cell technology, as well as our extensive experience with drug discovery and development, we wanted to, not only for COVID-19 expedite the clinical development of HSV 3013-2A, we also wanted to thinking about what type of patients is suitable for this kind of treatment. So we have our single cell technology can do immune profiling to identify patients most likely to respond. And we also thinking about next gen therapeutics. So among all the patients neutralizing antibody, we have identified, we select the most promising clinical candidate, move to clinical phase 1 trial. But in the meantime, we also have many antibodies, and some of them have been submitted to COVID efforts through Gates Foundation support. So we could think about multi-specific, which is actively ongoing right now, as well as combination cocktails, and to address different variants and next generation potential COVID variants that cannot be addressed by current efforts. But thinking even further, going beyond coronavirus, any other pandemic in the future, through this exercise, and we have seen, if we really putting our efforts together, we can advance rapid antibody discovery platforms through single cell technologies, through collaboration with academic and hospitals, through the industry collaboration with COVID. Such we would be really happy to position us into this part of the joint force to try to identify rapid responses to address any future pandemic as well. So with that, I'll be happy to take any questions.
Dr. Schweitzer, thank you so much. Very, very exciting talk. Thank you. And for the advances you're making in terms of therapy. I'm going to pose a question from the audience which came up. You addressed this briefly in your slide here. Mark Oldham is asking you, does your antibody, what's the specificity of your antibody? Does it bind to other coronaviruses, alpha, beta coronaviruses, MERS, etc.? And what are your strategies in thinking about those?
Yeah, that's an excellent question. So we did look at SARS and MERS, it doesn't. So where, based on the structure analysis, where we cover most of the surface between spike and ACE2. So this antibody is really specific for SARS-CoV-2. Then we have explored the possibilities. If you want it to be more pan across different species, you sometimes have to sacrifice the efficacy centered around SARS-CoV-2 just because the unique feature of SARS-CoV-2 and the ACE2 interaction. And where we think right now the most urgent needs to address is the SARS-CoV-2 and its mutant variants. So based on the structure, when the mutant happen at different locations, we think we can, some of the mutation will enhance the affinity towards the ACE2, but even so, because of the broader coverage of our interface, we actually could address. From that point of view, I think, you know, it could be, it's harder to design one molecule to address our needs. It's better to design an effective molecule to address our most pressing needs, then subsequently design other molecules to address evolving landscape. So that's the reason we're doing that. So in our antibody, we focused on, right now, this one is centered around S1 and the binding, but we also identified from patients antibodies targeted S2 for the fusion step. So you could imagine, you know, in the future when you have more broader pan, even others, maybe there's certain other steps, maybe more, you know, effective in the broader spectrum with different variants of other coronaviruses, different families. But right now, I think it's important to really zoom in on the most effective outcomes for this therapy.
Thank you. While the next speaker is loading up his slide, I'm going to ask you one more question. You know, what are the strategies Hi-Fi Bio is thinking in terms of delivery mechanisms for the antibody?
Right. So right now, for othe neutralizing antibody, the most quick, effective way is through IV, because that gives you the best bioavailability. And for us, just because we have prolonged half-life, we only have to do 1 times IV dose versus some other antibody you can see they need 2 or even multiple if you wanted to, you know, protect longer. So using the engineering for extended half-life actually also help us to address the delivery better. And we did intramuscular modeling for the distribution and bioavailability considered, where we actually think intramuscular with a smaller amount, if you know, administrate it shortly after infection, when the patient initially, you know, tested positive, or even before, if somebody have a high risk being exposed with family member, friends, you can get this intramuscular injection just 1 time. It's quite convenient. You can be protect up to a year without have to worry about injecting again. So the delivery is fairly straightforward.
Thank you. Thank you so much again for your very nice talk. I've now pleasure in introducing our next speaker.
Thank you.
Professor Neil King from the University of Washington Institute for Protein Design. He's a professor of biochemistry. I mean, he has pioneered development of new computational methods for designing novel assembling proteins. And we're really looking forward to his next generation strategies for vaccine development. I mean, in terms of protein design-based developments. So thank you. Welcome, Dr. King.
Thank you, and thanks so much for having me here today. I just want to make sure you can hear okay?
Yep, we can hear you well.
Excellent, all right.
And your slide is absolutely perfect.
Great. Okay, so I'd like to tell you a story about some of the technology development we've been doing here at the Institute for Protein Design. And it's really a story about technology platforms. As several of the previous speakers have mentioned, right, technology platforms have really come to the fore in terms of a rapid response to SARS-CoV-2. And so I'll tell you about one of the technology platforms we're developing here and go through a couple examples in what Bali called the BC, the before coronavirus era, and then finish up with application to SARS-CoV-2. Okay, so disclosure slide, we spun out a biotech company on some of this technology. So today what we'll be talking about are self-assembling or particulate immunogens or vaccines. And the reason we're talking about this is because particulate immunogens tend to generate or elicit much more robust immune responses than just small subunit vaccines, non-repetitive subunit vaccines. And as many of the speakers have discussed, subunit vaccines are becoming more and more prevalent in vaccine discovery and development because of their enhanced safety profiles, the ability to really engineer them for the optimal immune response, and things like that. This is an image on the left from a review from GSK showing, one of the mechanisms by which repetitive or particulate immunogens enhance immune responses. Here, it's really by cross-linking B cell receptors on the B cell surface, because this repetition, of course, is something that is a pathogen-associated molecular pattern. Viruses themselves are, particulate self-assemblies of proteins, and bacterial cell walls, of course, have repetitive chemical elements. So our immune system has evolved to detect repetition as a danger signal. And particulate immunogens really plug into that evolved feature of the immune system. It's not just B cell receptor cross-linking, however. Interactions with the innate immune system and also trafficking of these particulate immunogens in the body contribute to the enhanced responses they elicit. So of course, this is a vaccine modality with which we have lots of clinical experience. So the human papillomavirus and hepatitis B vaccines, of course, are self-assembling protein nanoparticles. So we have a long track record of safety and efficacy with this vaccine modality. And in fact, these two vaccines are two of the best subunit vaccines we have. So we know that self-assembling protein nanoparticles can be very, very good immunogens. So there was really what I consider to be a watershed paper that came from the VRC back in 2013. This is work from Masaru Kanekio, who at the time was in Gary Nabel's lab, but is now in Barney Graham's group. And they showed that this non-viral self-assembling protein, ferritin, which is an iron storage protein, was capable of scaffolding what I'll refer to as complex antigens. So this is, in this case, influenza hemagglutinin. It's a trimeric viral glycoprotein, a class 1 fusion protein, similar to SARS-CoV-2 spike. So what Masaru and his colleagues showed was that you can seamlessly integrate the antigen to your particulate display platform here, because these are both proteins, and so you just genetically fuse them. And when you produce this protein recombinantly in cells, out pop these little nanoparticles where every one looks just like every other, because, of course, the wonderful thing about proteins is that if there's a well-defined low-energy native state, anytime you make the protein, it will adopt that state. So these spiky particles displaying, in this case, 8 hemagglutinin trimers, in fact, did elicit superior humoral immune responses compared to the trivalent inactivated vaccine. So by 3 serological measures, they were much more potent than commercial vaccines. So this was really a beautiful paper because it showed us that non-viral self-assembling proteins, these assemblies that are stable enough, can accommodate these complex glycoprotein antigens. Since then, there's been an enormous amount of work exploring this concept. This is a very limited set of papers from this kind of subfield. And so it's really been remarkably successful, this concept of multivalent display on self-assembling proteins. However, the limitation is that when you're only using naturally occurring proteins, there's really a very small set of scaffolds at your disposal. So the 3 by far most commonly used ones are ferritin, encapsulin, and lumazine synthase. These account for the vast majority of papers in the field. And while wonderful particles, if these don't work for you, you're basically out of luck. Furthermore, these particles are not tolerant to substantial modification. So ferritin is ferritin, and it will always be ferritin. There's not really much you can do about it. And so if you want to make a bigger particle, for example, or a smaller one, or change the relative orientation of the antigen, you're just kind of out of luck. So one of the general concepts that underlies what we do at the Institute for Protein Design is we would like to build protein-based technologies that are tailored to specific applications. And so this is kind of a ridiculous example on the slide of what that might look like. If you want to build a new technology, let's say an alarm clock, and you only use materials that you have available around your house, you might come up with this ridiculous non-robust contraption. But this is not how human beings develop technologies. What we do is we make parts that are specifically suited to building the technology that we want. And when you do that, you come up with technologies that are simple, robust, and controllable. And so that's really what we're trying to do with proteins, is develop simple, robust, and controllable protein-based technologies. To do this, we use a software suite called Rosetta, which is the premier software suite for computational protein modeling and design. It was originally developed in the laboratory of David Baker, where I did my postdoc at UW, and is now being actively developed by a worldwide community of scientists. And so this is a graphical depiction of a method that we came up with in Rosetta for designing new self-assembling proteins. So new, for example, protein nanoparticles that you might use as vaccine scaffolds. So if we just focus on the top row here, this is really what the method looks like. It's two fundamental steps. There's a docking step and a design step. In the docking step, we build these materials the same way that Mother Nature does, which is through a combination of symmetry and non-covalent protein-protein interactions. If you think about all of the wonderful molecular machines in the cell, microtubules, ATP synthase, Rubisco, virus capsids, they're all built through a combination of symmetry and non-covalent protein-protein interactions. So we do the same thing. This happens to be icosahedral point group symmetry, which can be represented by 5-fold and 3-fold rotational axes. We go either find or build oligomeric protein building blocks that match those elements of symmetry in the target architecture, and we arrange them along the corresponding axes of symmetry and then dock them. We space them in and out and rotate them around their shared symmetry axes to find configurations where they fit together nicely. And then in the second step, we go in and we use Rosetta's sequence design capabilities to make up a new amino acid sequence at this novel protein-protein interface. And if we get it right, and this is the lowest energy state, then that interface, that designed interface, should drive assembly specifically to the target architecture. So what you get out of the computer is a hypothesis in 2 parts, a pair of amino acid sequences, and a 3-dimensional structure that you predict those amino acid sequences will adopt. So we take those hypotheses into the lab and test them, and this is what that looks like. So this is from a paper led by Jacob Bale, a very talented graduate student that I worked with a few years ago, where Jacob and I designed these 120 subunit megadalton scale assemblies with atomic level accuracy. So these things are about the size of an AAV, some of the smallest viruses. And you can see that for each of these 8 nanoparticles, where we got electron microscopy data on the left, you get just fields of monodispersed particles where every one looks like every other. And when you average those and compare them to projections calculated from the computational design model, you can see that they match quite closely. In a few cases, we were able to obtain crystal structures from these so that we could compare them on an atom by atom basis to what we made up in the computer. And we found that the deviation between what we made up in the computer and what occurs in the real world is less than an angstrom over all 120 subunits. So we designed these virus-sized complexes with atomic level accuracy. We predictively positioned every single atom right where we said it was going to go. And the implication of this, of course, is that you can now design customized structures. So if you want an architecture that's more porous, you might go for the one on the left of this slide. If you want an architecture that's less porous, you might go for the one on the right. If you want a 16 nanometer particle, a 17 nanometer particle, 18, 19, 20, you can design exactly that. What we're doing now, these days, is we're actually designing not only the particles, but the building blocks that make up the particle completely de novo. So this is recent work from George Ueda in collaboration with Alex Antanasievich in Andrew Ward's lab, Philip Brower and Rohir Sanders' lab, and several others, designing brand new monomeric proteins made up in the computer, and then designing those to form trimeric building blocks whose N-termini, represented by blue spheres in this image, are geometrically positioned for optimal genetic fusion to an antigen of interest. In this case, it's HIV envelope. So we, again, going back to kind of the Apple Watch, build the pieces for the technology you need analogy, right? We built a trimer specifically to scaffold HIV envelope and then use that to build particles. And when you do this, you can get nanoparticles presenting your antigen in a variety of predefined and customized geometries. So of course, we're using this as a platform for structure-based nanoparticle vaccine design, as well as for several other applications. And this is what it looks like for vaccines. So the main platform we're using, not all of them are like this, but the main platform we're using is what we call these two-component nanoparticles. So the nanoparticle itself is made from, in this case, a gray trimeric protein and an orange pentameric protein. We genetically fuse the antigen of interest, this is dsCAV1 from Barney and Peter Kwong, shown in blue, to one of the nanoparticle subunits, in this case, the trimer. So we're able to match the symmetry to the antigen. And then we express this as a standard recombinant biologic, nothing special about it, just like you would the antigen on its own. In a second expression host, which may be the same expression host or a different one, we often use E. coli because it's cheap, easy, and robust. We make the second nanoparticle component. We purify them independently, so the two proteins don't know that the other one exists until we mix them in a test tube, at which point they self-assemble to form these spiky particles. So I'm showing here a few examples in RSV, influenza, and HIV, but we've done this with a huge number of different antigens, of course, most recently with coronavirus S. This work has been in collaboration with a huge network of collaborators worldwide, and that's been one of the most fun parts of all this work, is working with all of these excellent virologists and immunologists to really work out this platform. So in the remaining time, I'm going to run through 3 examples rather quickly. And these 3 examples kind of highlight different aspects of the technology platform. So the first, and our first case was really RSV, where we found that multivalent display of prefusion F led to very potent neutralizing antibody responses. This was work that my group did in collaboration with Laurent Perez at the IRB in Switzerland and Karen Lore at the Karolinska. The second example will be in influenza, where we're working with Barney Graham and Masaru Kanekio at the VRC. And this is really a story about epitope focusing and improved breadth through multivalent display. And then the last story, of course, will be SARS-CoV-2. This is work my group has been doing in close collaboration with David Wiesler here at UW. And this is really a story about rapid platform deployment and ultra-potent neutralizing antibody responses. Okay, so RSV, we're just going to do a single slide. And this is just to show that multivalent presentation of prefusion F, this beautiful molecule that Barney introduced earlier today, induces a more potent humoral immune response than soluble antigens. This is very similar to what Masaru did with hemagglutinin back in 2013. And here what we're showing is that the nanoparticles give you a 10-fold higher neutralizing antibody response than the soluble trimer. Soluble trimer here already looks pretty good, and so this 10-fold increased response is really quite potent. We use the ratio of binding antibodies to neutralizing antibodies as a measure of antibody quality, and lower is better. And we find that the nanoparticles induce a higher quality, a more neutralizing antibody response than soluble antigen, and that the increased potency is strictly dependent upon nanoparticle assembly. So the open circles here are the same trimeric fusion protein that's in the nanoparticle, but a version of the pentameric, that orange pentamer subunit, that is incapable of driving particle assembly. So all of the proteins are there, all the T cell epitopes are there, the particle just doesn't assemble, and you get the exact same immune response as with soluble antigen. So the increased response that we're seeing here is directly linked to nanoparticle formation. Okay, second example is on influenza hemagglutinin. This work is up on bioRxiv, and we just submitted the revision, so it should be out soon. And here what we're doing is we're working with Barney and Masaru to put multiple different hemagglutinins either onto multiple distinct nanoparticles or co-display them all on the same nanoparticle. And this mosaic nanoparticle is really made possible by this 2-component nanoparticle technology, where you can make 4 different hemagglutinin I53D and 5B fusions. D and 5B is just the nanoparticle subunit in this case. You mix those 4 trimers together and nothing happens, and then you spike in the pentamer and you get assembly of this mosaic immunogen. These nanoparticles are, again, very monodisperse, that every particle looks like every other, including for the mosaic, which we showed antigenically and by mass spec comprises all 4 antigens in the intended ratio. So you really have a lot of control over which antigens you're co-displaying on these particles. When we take these into animals, we find that when we compare either the cocktail vaccine in blue, the 4 different particles mixed together, or the mosaic nanoparticle co-displaying HA in pink, to commercial vaccines, this is quadrivalent inactivated or influenza vaccine, we find that against vaccine strains, so the same viruses whose hemagglutinins appear in the particle, we get immune responses, including HAI, that are equivalent or maybe slightly superior to current commercial vaccines. And this is really important from a regulatory perspective, but being as good as commercial vaccines is maybe not that exciting. What we're really excited about is that when you look at distant cousins on the flu family tree, so these are heterosubtypic viruses, you know, H5N1 bird flu or H7N9 flu, this is where you see the nanoparticle vaccines, both of them, significantly outperform commercial vaccines, which give you very poor heterosubtypic responses. This is manifested as well in protection studies where both nanoparticle vaccines provide very good protection against heterosubtypic challenge, whereas current commercial vaccines perform very poorly in the same experiment. For the NHPs, we didn't challenge the NHPs, but we showed that there are protective antibodies elicited in these NHPs using passive transfer experiments to mice, where mice receiving polyclonal Ig from the NHPs, again, were protected against heterosubtypic challenge, showing that this really is antibody mediated. And you'll notice here that while both nanoparticle vaccines do very well, the mosaic, where you're co-displaying multiple hemagglutinins, seems to subtly yet consistently outperform the cocktail. And we've seen that across all of our studies so far. Again, the enhanced responses here are directly linked to nanoparticle formation. We did the same thing where we reverted the computationally designed interface back to the wild type residues here so that these trimers can no longer assemble. And what we saw in that case was that while vaccine strain responses were maintained, so the yellow group is now the non-assembling control, these heterosubtypic responses, which are stem-directed, were totally lost. And we dropped back down to the level of commercial vaccines. So here, what we're really seeing is nanoparticle display doesn't necessarily give you increased potency against the vaccine strain, but it does seem to redirect the response against the stem epitope and give you improved breadth. We wanted to dig into that a little more, so we did some serological experiments showing that indeed we are getting improved stem-directed responses. I won't go through this in detail, but that's what those data show. And then David Wiesler here in the biochemistry department at UW did polyclonal EM, this recently developed technique out of Andrew Ward's lab, where you can process Fabs from polyclonal sera. So there's a huge number of different Fabs with different specificities in the sample. You make complexes with your antigens, you purify them and visualize them by EM, and then let the software classify out the different types of antibodies that you're getting and really map the antibody response and the epitope specificity visually. And what we found was that with either the cocktail or the mosaic nanoparticle vaccines, when we looked at strain matched responses, so this is against, you know, an H1 hemagglutinin Michigan 15 that's in the vaccine, we got a lot of head-directed antibodies. So, you know, the vast majority of the antibodies were directed against the receptor binding domain or the vestigial esterase domain, both up here in the head. And we got a very small, although observable, number of stem-directed Fabs, like this one you can see elicited by the mosaic group. However, when we formed complexes with a heterologous antigen, so this is H5HA from an H5N1 virus, we saw only stem-directed Fabs, showing that this heterosubtypic response that we're getting and this broadly protective response that we're getting is entirely stem-directed. And again, you don't get this from the commercial vaccines. This class here, you can see there are actually at least 2 different classes elicited. You can notice the different angle of rotation in these 2 Fabs. This one actually looks an awful lot like Medi8852, which is a human BNAb. And so this suggests that we're eliciting these BNAb-like antibodies through display on this nanoparticle platform. Okay, so switching to our last story, the topic of today is SARS-CoV-2. Again, this is really a story about deploying a technology platform. And so we had actually been working on coronavirus nanoparticle vaccines, both with Barney Graham and with David Wiesler since about 2017. And so we had done some previous work in this area. And so when SARS-CoV-2 broke out, we immediately went and put the S2P trimer, the prefusion stabilized form that Barney developed in collaboration with Andrew Ward and Jason McClellan, on our nanoparticles. And we can make beautiful particles with the S2P trimer on there. And the concept behind this immunogen is really that, you know, by putting the whole trimer on there, including S2, the idea is that there may be cross-reactive epitopes beyond just the strain-specific epitopes that are present in the RBD. However, you know, our previous data with SARS and MERS and other coronaviruses showed that multivalent display of S2P didn't really improve the immune response. And also the S2P trimer is really, you know, making these proteins is still a challenge. The yield is fairly low, much better than wild type, but still fairly low, and they're much less stable than the RBD. So kind of as a backup, David Wiesler and I decided to put just the receptor binding domain of SARS-CoV-2 spike on the particle. Again, we can make beautiful particles. The concept behind this immunogen is that, you know, the vast majority of neutralizing activity in infected humans is directed at the RBD. That includes potent receptor blocking antibodies. And so this is really, it seems like for a potent neutralizing response, it seems like this is the part of the molecule that you really want to hit. So we're trying to focus the response on these potently neutralizing epitopes. So what we found is that multivalent display of the RBD makes it much more immunogenic than it is on its own. And these particles, as I'll show you, are very manufacturable. You get a ton of protein. It's really easy to make. It's very, very stable. So I'm only going to talk today about RBD I5350. We're continuing to explore display of the prefusion stabilized trimer, but I won't talk about that today. So this is just some data from our recent publication on this. Again, we're taking the RBD out of spike. The RBD has 2 N-linked glycans. Both are present in our vaccine because we're making this component in mammalian cells, so CHO for large-scale production, and we're making the orange pentamer again in E. coli. You purify these 2 recombinant biologics, you mix them, and you get these particles. Particles are very monodisperse, as evidenced by light scattering and of course, negative stain EM. And then we looked at the stability of these things, so we went out to 28 days at 3 different temperatures, including basically room temp, 22 to 27 degrees. And we found that the components, we made a few different linker variants of these particles. All of those, as well as the particles themselves, are very, very stable out to at least 28 days. Additional stability studies are ongoing. They maintain their antigenicity. They don't aggregate. And when you look at particles that have been sitting at 22 to 27 degrees for a month, they're indistinguishable from particles that have been sitting in the minus 80. So they're very, very stable. And then of course, what we really want to see is the immunogenicity. So this is an experiment in wild type mice. After 2 immunizations of either 0.9 micrograms or 5 micrograms of antigen, we can see that, you know, post-prime, the particles are already inducing neutralizing antibody responses that are comparable to human convalescent sera, whereas the monomeric RBD formulated with Adavax or the soluble S2P trimer formulated with Adavax in this stringent pseudovirus neutralization assay don't give us any neutralization. After the second immunization, we're starting to see some neutralizing activity from the S2P trimer that is, you know, on par with human convalescent sera. This is in line with results that other people have obtained with recombinant S2P trimer. But the nanoparticles are inducing neutralizing antibody responses that are 1 to 2 orders of magnitude more potent. And when we take this into challenge studies, we did this in collaboration with Ralph Barrick at the University of North Carolina using their SARS-CoV-2 MA virus. We find that all of the nanoparticle vaccines provide full protection in both the lung and the nose, whereas we do start to see breakthrough from the S2P trimer at low dose, and the monomeric RBD even at high dose fails to protect at all. And the last point I want to make on the SARS-CoV-2 RBD nanoparticle vaccines is, you know, you might suspect, oh, well, with the RBD, maybe there's an increased risk for viral escape through mutation. However, there are multiple independent non-overlapping epitopes in the RBD that lead to neutralization. And we did serum competition assays using polyclonal sera that we obtained from immunized NHPs, and showed that in all 3 of these tested epitopes, we're getting significant competition with the polyclonal sera. So this vaccine is eliciting polyclonal responses of multiple specificities, at least 3 independent neutralizing epitopes, and which really should minimize the risk of viral escape through mutation. So this vaccine is on its way. We've tech transferred it out to 2 different manufacturers, one in the US and one in South Korea. GMP harvest has happened recently, and the particles are on their way to clinical trials, the first of which we're on track to begin next month before the end of the year. We're doing a large challenge study in NHPs with Bali Palindran and his group, and his talented postdoc Prabhu that he mentioned. The data are starting to come back there, and they confirm the very high potency of the RBD nanoparticle vaccine and the protection that it affords in NHPs. We are also working with new pre-fusion stabilized S ectodomains. So this is specifically Hexapro from Jason McClellan's group. And when we display that on the particles, we're starting to see enhanced stability and immunogenicity. So I think there's still room to explore the display of the entire S trimer on the particle. And one of the things we're doing there is we're really now going back and designing new nanoparticles that are specifically optimized for eliciting the best immune response against the S ectodomain trimer. And it's something that, you know, you really couldn't do any other way except through computational protein design. So we're very excited about the after coronavirus vaccinology that Bali mentioned. So obviously these efforts have been hugely collaborative, much of which was really kind of set up and driven by the Bill and Melinda Gates Foundation. So I just think what they're doing this year in SARS-CoV-2 vaccines is absolutely amazing. So here are the people from my group that have supported this work. And then we have what we call these core laboratories at the IEPD, including a nanoparticle core laboratory. And our SARS-CoV-2 project was really led by the cores. And this team of people really came together in an amazing way and pushed things forward. SARS-CoV-2 work, of course, is in close collaboration with David Wiesler and Bali Palindran. HIV with Andrew Ward and Rohir Sanders, flu and coronavirus work with Barney Graham and Masaru Kanakiyo. And then Ralph Barrick was really instrumental for live virus neutralization and challenge studies. So thanks again for having me here, and I'd be delighted to take any questions.
Neil, I mean, absolutely futuristic and fantastic talk. You know, thank you. I mean, the brave new world with new ways of doing things, fantastic. Thank you. I mean, I have a couple of questions for you. One is that in terms of your designed, scaffolds, do they induce specific conformational change in these immunogens, for example, in S2P, or do they stabilize, or do they alter the ability for a conformational landscape for these proteins?
Right, that's a great question. Thank you. So actually, we go to great pains to make sure that they don't, right? And the best tools we have for that are really monoclonal antibodies. So we characterize them antigenically and show that all of the monoclonal antibodies bind in the right way. I kind of glossed over it, but on the first slide on influenza, David Wiesler's group was actually able to obtain a localized reconstruction of the displayed hemagglutinin at 3.3 angstroms resolution. So they got a structure of hemagglutinin on the particle, and it looks perfect. There's no difference at all. So, you know, the improvements in cryo-EM are letting us look at this now too in great, great detail. The one thing I will say on this topic though, is we've noticed in several instances that fusion of these glycoprotein antigens to the nanoparticles actually physically stabilizes them. So it can further stabilize the pre-fusion form, for example.
Thank you. The other question is, your designed protein engineered nanoparticle, does it also elicit additional immune response by itself, which you're observing in terms of looking at the cellular immune system?
So you know, the protein scaffold is a foreign protein that is symmetric, right, and repetitive, and so you absolutely get a response against it. You get antibodies against the scaffold. We always measure those and include them in our publications, so we're very straight up front about that. The next question is, does that have any effect on the antigen-specific response? In the experiments that we've done to date, the answer is no. So if we pre-immunize animals with just the nanoparticle but no antigen, and then we come in and boost those animals with the nanoparticle with the antigen on it, the antigen-specific response is just as potent as in animals that have no pre-existing immunity. So that's one experiment.
But the non-specific immune response, response to your nanoparticle, is that going to be deleterious for the subject in any way?
Yeah, I mean, I don't think we're really going to know until we go into humans. So all three of the programs that I discussed, RSV, flu, and SARS-CoV-2, will be in phase 1 clinical trials within the next 6 months. So we're about to start getting a lot of data in humans on this. You know, they're proteins. We don't expect any abnormal responses to them. People that have put ferritin nanoparticles, for example, in humans, Barney's done a ton of this, see no weird or deleterious responses to ferritin. So we don't expect to see anything, but we'll obviously need to closely monitor that as these programs progress.
Thank you so much, Neil. We have questions on the Q&A. If you don't mind, you can feel free to respond later on to them, and they can communicate with you. In the interest of time, I would like to go to our, we're talking very nicely about biochemical, chemical, cellular interventions. How about digital therapeutics? In fact, digital therapeutics, which is very important, is the most, it's an exciting topic for us. We are going to conclude this with our COVID Forum 4th session with a very exciting talk on digital therapeutics. We have, I have tremendous pleasure in welcoming Beth Jaworski, who is in the U.S. Department of Veterans Affairs, and she's the lead for the Mobile UX Research VA National Center for PTSD Dissemination and Training Division. Her research focuses on how mobile health apps can be designed to promote engagement, increase mental health literacy, and help mental health disparities. She has a doctorate in social psychology from UC Santa Cruz. I think in times like this, mental health is one of the most crucial issues, and digital therapeutics would probably be the best way to deal with it. So Beth, we're really looking forward to your talk to help us be sane for the future. And thank you and welcome to this session, and look forward to your presentation.
Hi, thank you so much. It really is an honor to be invited to speak today among such talented scientists and researchers. Today I'll be speaking about the COVID Coach app and digital strategies for addressing the mental health impacts of COVID-19. I'd like to begin by thanking the incredible team that has been instrumental in this work, including my colleagues within the VA's National Center for PTSD, as well as our technical partners at Vertical and Mumble. During the previous talks, you heard about the phenomenal work that has been undertaken in developing a vaccine for the coronavirus. I'd like to shift gears a bit and highlight the need for addressing the mental health impact of the pandemic. Back in May, the United Nations released a policy brief and noted that although the COVID-19 crisis is, in the first instance, a physical health crisis, it has the seeds of a major mental health crisis as well. If action is not taken, good mental health is critical to the functioning of society at the best of times. It must be front and center of every country's response to and recovery from the COVID-19 pandemic. The mental health and well-being of whole societies have been severely impacted by this crisis and are a priority to be addressed urgently. I think this statement continues to be incredibly accurate, and if anything, the sense of urgency for finding ways to address the mental health impact has grown. This is likely not surprising to anyone here today, but there's a rapidly growing body of evidence indicating the pandemic is having a negative impact on mental health globally. For example, among adults in the US, the Centers for Disease Control and Prevention found that over 40% of survey respondents during a survey conducted in June reported at least one adverse mental or behavioral health condition, including symptoms of depression, anxiety, post-traumatic stress, or having started or increased substance use to cope with stress or emotions related to COVID-19. Over 10% of respondents reported seriously considering suicide in the past 30 days, and the rates for these reports were significantly higher among young adults, Black and Latinx populations, unpaid caregivers, and essential workers. As Dr. Torres mentioned in his talk on Thursday, a very recent study published in The Lancet indicates a bidirectional relationship between psychiatric disorders and COVID-19. So having a psychiatric disorder is associated with a greater likelihood of getting COVID-19, and getting COVID-19 is associated with an increased risk of receiving a psychiatric diagnosis 14 to 90 days post-COVID-19 diagnosis. And I think importantly, we don't know the long-term psychological impacts and consequences of all the stress, fear, loss, grief, and uncertainty due to the pandemic. So very broadly, why are digital mental health resources one way to address the psychological impacts of COVID-19? One primary reason is because they can be used to increase remote access to resources. There's a shortage of mental health professionals. Not everyone needs to see a professional for subthreshold levels of disorders, and many people can benefit from self-management tools regardless of whether they are receiving professional care. Options that don't require in-person or face-to-face contact are needed due to the nature of COVID-19 transmission. Digital mental health resources can be scalable, so the potential for reach is great. For example, in the US, the majority of individuals own smartphones, even among low socioeconomic status groups. Digital mental health resources may also help alleviate some of the barriers to accessing care. So for example, being able to connect with resources without needing to travel anywhere. And they can be useful as a resource for a range of mental health concerns, including anxiety and depression and PTSD. There are many different types of digital health interventions and many different approaches to understanding their impact. Today, I'll primarily be focusing on the use of apps from a public mental health perspective. The VA's National Center for PTSD has been in the digital public mental health space for nearly a decade, with a focus on creating publicly available mobile mental health apps. PTSD Coach, the first app created by the team, was released in 2011, and the portfolio continues to grow. These apps support mental health self-management for PTSD and related concerns, as well as treatment companion apps designed to support evidence-based psychotherapies, such as prolonged exposure therapy for post-traumatic stress disorder and CBTI Coach for cognitive behavioral therapy for insomnia. In total, all the apps in the portfolio have been downloaded over 4.5 million times. Data privacy protections are essential. No personal identifiers are ever collected. The apps are all free, and once downloaded, Wi-Fi or a data plan is not needed to access the vast majority of the content. They are also all fully accessible, meaning they are compatible with assistive software technologies like VoiceOver and TalkBack for those with vision impairments. And there is a growing body of evidence to support that these apps can be helpful when used independently. So why did we make COVID Coach? We began work on COVID Coach late in March based on a few key factors. We have been following the research coming out of China and Italy, noting the increasing mental health concerns associated with the pandemic. We began receiving requests from VA providers for resources to help support the rapid transition to telemental health as shelter-in-place orders began taking effect. And as a team, we felt strongly about contributing to the COVID-19 crisis as part of the VA's fourth mission, which is to help the nation during times of national emergencies and disasters. We decided to create an app that drew on many of the best aspects of our existing products, primarily from the PTSD Coach app, but focused on the unique challenges of the pandemic. We created COVID Coach to provide app users with interactive, evidence-informed tools for coping with stress and anxiety, information about how to stay well, stay connected, and navigate pandemic-related challenges, self-monitoring of goals and mental health symptoms, and reputable resources to discover and connect with various types of help and support. It was not designed to treat any mental health condition, and it is not a replacement for professional care. I would like to provide a brief overview of the key content areas because I'll be sharing some very preliminary data analyses related to how app users have been engaging with each of the four main sections that you can see on the home screen of the app pictured here. The Manage Stress section in the upper left quadrant of the screenshot, the Learn section, the Mood Check section, and Find Resources. In the Manage Stress section, you can select a challenge you'd like help facing, and the app recommends a tool, or you can choose the tool directly. There are 57 different tools to choose from. These tools are based on cognitive behavioral strategies and techniques, as well as other evidence-informed strategies for stress management, such as mindfulness. And you can also create a list of favorite tools. Here's one example of a tool, the audio-guided tool called Deep Breathing. The Learn content for COVID Coach includes 5 major sections and 57 unique topics. Staying Well draws on disaster mental health and focuses on psychological well-being and managing emotions during the pandemic. Staying Balanced, which features content for helping people navigate the unknown or unexpected, like job loss. Staying Together focuses on facing challenges related to different types of relationships, such as with partners, roommates, or children. Staying Safe offers information related to intimate partner violence, child abuse and neglect, problem drinking, suicidality, and xenophobia and racism. And Staying Healthy focuses on staying healthy from COVID-19 infection, drawing on the CDC, World Health Organization, and other public health information. The mood check section includes goal setting and assessments to track well-being, as well as symptoms of anxiety, depression, and PTSD using validated scales. Here's an example of the goal setting sequence. You can create your own goals, rate your success in accomplishing them, and view your progress over time. And here's an example of a question from the assessment for tracking symptoms of depression, what the feedback looks like, including total score and links to crisis resources, and how the data are visualized for users. And lastly, the Find Resources section includes a comprehensive range of services and supports that individuals can access, including crisis support and how to meet basic needs, like how to find help for paying bills. In order to understand how app users are engaging with the app, we have a custom app instrumentation solution that our technical partners have developed. Because we cannot collect any personal identifiers, including device ID, we're not able to use any of the off-the-shelf instrumentation and analytics solutions. We do not require users to create an account, and the only way that we are able to track usage is by creating a custom identifier, which we call an install code. When an app user installs the app and then opens it on their device for the first time, a randomly seeded alphanumeric string is generated using a custom method and passed to the MD5 hash function. We capture all in-app events with the exception of any free text entry. So we have the possibility for very detailed analyses of how people are using the apps, but we lack information about who is using the apps. Here's a screenshot from the custom app dashboard for COVID Coach, just so you can see what it looks like. So we are able to view, filter, and extract event level app data. So what do we know so far about how many people have downloaded the app and what they think about it? We know that since the app was released during the third week of April 2020, it has been downloaded over 140,000 times. Google is currently making it difficult to find apps related to COVID-19, and we have yet to be able to have them include COVID Coach among the recommended apps for COVID-19. In order to find it in the Play Store, you need to have the direct download link or search for COVID Coach as a single word. So you can see that challenge reflected in the download numbers for Android compared to iOS. Although star ratings have their limitations, it is one way for us to gauge what the community thinks about the app. In this case, it has been well received. And within the menu section of the app, you can opt in to an anonymous 12-item UX feedback questionnaire, like the screenshot on the side here. It covers topics such as how users learned about the app, what they think of the design, why they use the app, what sections they use the most and least, and if they would recommend it. And so far, nearly all respondents indicated that they would recommend the app to a family member or friend. I'd now like to present some very preliminary data about COVID Coach as a case study and way to demonstrate the possibility and potential of digital mental health interventions to support mental health and well-being during the pandemic. I focused on the time period of May 1st through October 31st, 2020. In that window, there were nearly 3.4 million app events. This includes every screen view and every button press, and the total number of unique app users, as approximated by the install codes that I described earlier, was nearly 50,000 in that 6-month window. In terms of daily active users, we saw a spike in early May due to a VA marketing push where COVID Coach was featured in an email that went out to about 12 million people, as well as being featured in Wired magazine. Since May, usage has leveled off but remains steady. 48.6% of app users only use the app on one day, but there is some variability. Much like many mobile mental health interventions, there is certainly room for improvement with respect to engagement. The app contains 4 main sections, and this table is focused on a few key events within those sections, including using a tool within the Manage Stress section, viewing a Learn topic, creating and submitting a goal rating or completing at least one of the 4 assessments, and viewing a resource. The Manage Stress tools were the most used, as well as used by the most number of people. Managed stress tool usage accounted for over 70% of these key app events. However, even among the lesser completed events, there were still thousands of actions across thousands of app users. To explore the usage of this key content from a different angle, this is a graph of the mean number of unique and total tools used among all users who tried at least one tool compared to the mean, unique, and total number of learned topics viewed by app users who viewed at least one learned topic. So these numbers suggest that on average, app users who try tools are trying more unique tools and using more tools overall compared to app users who view learned content. For the learned content, because the means for unique and total learned topics viewed are much closer together, it suggests that users may be viewing learned topics only once and not returning to reread them. Among all the 57 tools in the app, the audio-guided exercises are most popular, and 4 out of the top 5 tools are some of the audio-guided ones, including the deep breathing exercise I showed previously. The Change Your Perspective tool, shown in the screenshot, is a tool designed to help people shift or reframe their thinking in a more helpful way. The Find Resource section was the least used among the 4 key content areas, but I think it's important to note that although it was least used relative to the other key content areas, it still represents thousands of views by nearly 10,000 unique app users. Finding local resources was the most visited section, which is where users can select their state and find state-specific information about COVID-19. And the Meeting Your Needs section is also important to highlight, as that is the section that contains information about meeting basic needs, such as how to find local food banks and how to apply for benefits like unemployment and temporary assistance for needy families. And lastly, although the Crisis Resources section was not the most frequently visited, it was viewed over 3,000 times by over 2,000 unique users. And these are resources that link to the Suicide Prevention Hotline, Domestic Abuse Violence Hotline, the Trevor Project, those types of resources. I'd now like to turn to the key events within the mood check section of the app. Not every app user completed assessments within this section, but I think there are some important trends to note based on the people who did complete these assessments. To begin with, I'd like to highlight well-being. The mean scores on this graph are for the first well-being assessment score among app users who completed the well-being assessment on their first day of app use. The well-being scale is scored from 14 to 70, where higher scores indicate greater levels of well-being. A score below 47 is indicative of low well-being. So all of the mean scores here by month fell below that cut point, and you can see a slight downward trend month over month within this 6-month window. With respect to understanding if the app is being used by individuals potentially experiencing mental health issues, this graph highlights that COVID Coach appears to be reaching individuals experiencing symptoms of depression. This is a graph of the distribution of the first PHQ-9 score among individuals that completed a PHQ-9 on their first day of using the app. Based on the cut point scores, the majority of users who completed the PHQ-9 appear to be experiencing some level of depression symptoms, with only about 16% of users falling into the minimal range for symptom severity. So you can see that the majority of the users that completed this assessment are falling into the symptom severity categories of mild, moderate, moderately severe, and severe. Taking the same population of app users from the last slide, individuals that completed their first PHQ-9 on their first day of app use, I examined mean app usage patterns by the symptom severity categories. So here you can see a pattern that's consistent with overall usage patterns, meaning that tools are the most used section of the app, but you can also see that there is a trend in the usage such that individuals with mild, moderate, moderately severe, and severe depression symptoms appear to be using the app for more days on average, and on average are using a greater number of tools than individuals with minimal depression symptoms. Again, these are just preliminary analyses, and I'm noting the trends and patterns. I'd now like to talk about symptoms of anxiety. And so we are seeing similar trends with respect to symptoms of anxiety. This is a graph of the distribution of the first GAD-7 score among individuals that completed a GAD-7 on their first day of using the app. Based on the cut point scores, the majority of users who completed the GAD-7 appear to be experiencing some level of anxiety symptoms, with only about 13% of users falling into the minimal range. And again, taking the same population of app users from the previous slide, individuals that completed their first G87 on their first day of app use, I looked at the mean app usage patterns by symptom severity categories for anxiety. And here you can see a pattern that's consistent with overall usage patterns and consistent with what you just saw for symptoms of depression. However, there appears to be a slight difference in that individuals with mild, moderate, and severe anxiety symptoms appear to be using the app for more days than those with minimal symptoms, but those with severe anxiety seem to be using slightly fewer tools on average than individuals in all other symptom severity categories. To summarize these preliminary findings for COVID Coach, many people are using the app and seem to like it. Usage spiked in May, but has remained steady. Although many users only try the app once, there is a lot of variability. Managed stress is the most used key content area, but there are thousands of users accessing thousands of different resources and tracking mental health symptoms. The app does appear to be reaching individuals with varying levels of distress, including people with symptoms of depression and anxiety. And usage trends suggest those with mild and moderate symptoms may be using the app more than those with minimal and severe symptoms, which is consistent with previous research in this area. I'd like to end by sharing just a few thoughts about how I believe we can contribute to advancing the reach and impact of digital mental health resources during the pandemic and beyond. I think that this is a really important moment in time for digital mental health interventions. There's been a push to be creative and innovative and to bring together diverse teams of experts to develop solutions because there is such a great need for resources that can be used remotely. However, because of the widespread and well-publicized mental health consequences of the pandemic, this might also be a unique opportunity to amplify efforts to destigmatize mental health. This is something that could be directly incorporated into the digital mental health products themselves, as well as the marketing of them. With respect to apps in particular, they're certainly not the only way to address mental health concerns, but I do think they have a role to play as part of a multi-pronged public mental health approach because they are a scalable way to disseminate accurate information and resources, and they can be tailored to meet the needs of certain groups or individuals. But the design should reflect what we know about how and why people use apps. For self-management apps in particular, they need to be brief, engaging, and easy to use. What we've learned so far from COVID Coach is that there is room for improvement, particularly around engagement. Institutional support and dissemination efforts are key. Building great digital tools doesn't mean that people will find them or use them. So more work is needed to raise awareness and help people learn about digital mental health resources that are currently available and how they can use them. And lastly, often stated, but I think certainly true here, more research is needed to better understand for whom, why, and under what conditions digital mental health interventions may be most helpful. And in particular, focusing on the questions that center the needs of the underserved populations and shed light on how to optimize interventions to help alleviate mental health disparities. In sum, I'm hopeful that digital health strategies can help address the mental health impacts of the pandemic, and I'm very much looking forward to seeing how the science and innovation in this area can be used to drive mental health access and equity in the future. And I would like to say thank you so much. And here's the contact information for myself, as well as the mobile mental health team at the National Center for PTSD.
Thank you, Beth. I mean, really great talk. Thank you very much for your talk. And I mean, before I turn it over back to Professor Akai for concluding remarks, let me pose the question that was given by Mark Oldham. It's a bit long, complicated question. Is the increase in mental health issues post-disease associated with COVID patients further associated with long haulers and or more specifically with measurable brain damage? That's one part of his question. The other part is the increased likelihood of contracting COVID associated with mental disease further associated with differences in behavior such as disregarding guidelines as a result of mental disease?
That is a great question. I'm not sure that I have the answer to it. I think that the research unpacking these the causality of these relationships is not quite there yet. I suspect, that there probably are relationships, or at least associations, between people's beliefs about the severity and the importance of mask wearing, that sort of thing, and their engagement in behaviors that could prevent the transmission are certainly related to outcomes there. And it may be very well the case that individuals with mental illness have less understanding about the need and the consequences for not engaging in protective behaviors. So I don't have a great answer to that question, but I think it's a very good one.
Yeah, thank you. It's a complex question. So, I just want to turn it over now to Dr. Metin Akai. And I'm getting comments that it's a great talk and great. It's a great session, all 4 sessions, and especially today's session. I will now turn it to Professor Akai for concluding remarks and comments. Thank you.
Thank you so much, Professor Shankar Subramanian. First of all, on behalf of the IEEE Engineering Medicine and Biology, I would like to appreciate all these 29 exceptional speakers. Talks were highly inspiring, highly insightful, highly instructive. And also, I would like to express my gratitude to Professor Shankar Subramanian, Paolo Bonato, and Colin Brennan. They were the 3 organizers with me to put this forum together. I would like to also thank Mike Markowitz, Dr. Ting Cheng, Nick Plasko, and Knati Konun, as well as Dr. Charlotte Swaits. Their helps were tremendous and highly appreciated. We are technical society. We are IEEE EMBS. Our mission is to promote, foster healthcare innovations and excellency for the well-beings of our community globally. We are here to develop technologies, and also the goal is to increase the quality of the healthcare, lower the healthcare cost. The way to do it, our mission is to build communities. The way to do it, our mission is to structure and build a platform. That way, the people, they can exchange ideas, and disseminate their information, knowledge. This way, we create next generation leaders. This forum was very important. It was the eye-opening in a way that is, we have 4 areas covered: healthcare, screening, tracing, and treatment in the forum. We have a lot of exceptional conferences, workshops, but I believe that this is one of the most unique and comprehensive one to have all these 3 interesting areas. For futures, we plan to have several more. The forums, Grand Challenges Forum, to continue, increase the awareness of emerging technologies, continue building platforms, continue building communities. One is going to be on data science. We decided to have it last year with the collaboration with IEEE EMBS Technion under the leadership of Dan Adams, Paul Sajda, myself, and Dr. Ming Po, Dr. Bonato. Unfortunately, because of the pandemics, we were unable to have it in Technion campus May 2020. We are going to have it, and we will have it first virtual, then we will have face-to-face. We will go back to Israel. We will also have other inspiring forums, including technology for optometry, will be led by Dr. Jim Whalen. And I'm also hoping that Dr. Shankar Subramanian will lead the systems medicine and systems biology the forum in the incoming years. So this is to finalize, as we have several the activities as a society. Next one, we are going to have our signature technical conference on neural engineering, first week of May 2021. Unfortunately, it will be also a virtual one. That effort will be led Silvestro Michera, Thomas Stiglitz, Paolo Bonato, and myself. We are going to have this conference in Italy 2023, and with the 2 years delay. Our main conference, flagship conference, will be held Guadalajara in Mexico. Guadalajara is right now is the hub for the innovation, hub for mid and small size companies. I think we are going to have very unique conference. As of today, IEEE Executive Board, EMBS Executive Board have been discussing the plans and whether or not to have it virtual or hybrid, and the decision will be conveyed to our community from the Executive Office of the IEEE EMBS. We are going to have another, the signature, the special topic conference on the BHI, Biomedical Health Informatics, will be led Dimitris Fotiadis. It will be held in Greece. As you can see that we have technically, we are building the conferences, and our journals are also extremely attracting a lot of wonderful papers, and we are doing great. Our impact factors are increasing. Among them, a BHI impact factor is right now is the top, among the top impact factor among the several biomedical informatics related journal, flagship journal, the Transaction Biomedical Engineering, and our new journal, Open Access Journal, led by Paolo Bonato, is attracting exceptional papers. So the conclusion, we appreciate your time, we appreciate your participation, and we will continue, move ahead, and we will continue and advancing the technologies for the benefit of the people. The final note, Dr. Professor Fukuda, who is the president of the IEEE, he wanted to join and welcome all of you at the beginning of the forum. Regrettably, because of the technical difficulties, he was able to connect late, and also time was extremely late in Tokyo. He asked me to convey his message. We, IEEE Technology Society, we are the largest international, largest scientific organization in the world with more than 420,000 members. We are here to advance the technology for humanity of the people. Thank you so much, and please stay safe, and we will be in touch. Have a nice day.